A single photon strikes a leaf, and in that instant, faster than chemistry should allow, it finds its way through a maze of molecules, taking every possible path at once, as if the plant itself were dreaming the light into being. There is something quietly astonishing in this, the idea that the green world outside the window operates at the edge of physics, where particles behave like waves and energy moves through quantum corridors too small to imagine and too elegant to ignore. If these slow journeys through science bring a kind of stillness, subscribing means more will arrive when they are needed. The story of how leaves capture sunlight begins not with what we can see, but with what we can barely measure, photons entering the architecture of a chloroplast, stepping into a dance that has been perfected across 3 billion years of quiet evolution, and this is where we begin. Let us begin now with a story, and it is the sort of story that unfolds slowly, as all the best ones do, beginning not with any grand announcement or moment of sudden clarity, but rather with the patient accumulation of observations across decades, the careful work of scientists who could not have known, as they peered into their instruments and recorded their measurements, that they were laying the foundations for something quite remarkable. This is the story of how we came to understand that plants, and certain ancient bacteria, may be doing something that physicists once thought impossible at the warm, wet, bustling conditions of living systems, and it begins, as many stories in science do, with someone noticing something curious and deciding to look more closely. The Fenner-Matthews-Olson complex, which we shall come to know rather well in the minutes ahead, takes its name from 3 scientists whose work spans nearly half a century, and the first of these names belongs to John Olson, who was working at Brookhaven National Laboratory on Long Island in New York during the 1960s, at a time when the molecular machinery of photosynthesis was still largely mysterious. Olson was studying green sulfur bacteria, ancient organisms that thrive in conditions that would seem impossibly dim to most photosynthetic life, making their living in the depths of lakes and in the mud of tidal flats where only the faintest whispers of light penetrate. These bacteria had evolved, over billions of years, to become extraordinarily efficient at capturing whatever scarce photons came their way, and Olson suspected that understanding their light-harvesting apparatus might reveal something fundamental about how nature manages this remarkable feat. During his time at Brookhaven, Olson worked to isolate and characterize the proteins involved in the early steps of photosynthesis in these green sulfur bacteria, particularly a species called Chlorobium limicola, and his careful biochemical work established that there existed, within these organisms, a distinct protein complex that served as a kind of energetic bridge, accepting the energy captured by the large antenna systems and funneling it, with what seemed like remarkable precision, toward the reaction center where the actual chemistry of photosynthesis takes place. This was painstaking work, the sort of quiet laboratory effort that rarely makes headlines but which forms the bedrock upon which later discoveries are built, and Olson's contributions during this period established the basic outlines of the energy transfer pathway in these fascinating organisms. A decade later, in 1975, Roger Fenner and Brian Matthews achieved something that had eluded researchers until that point, which was to determine the precise three-dimensional structure of this energy transfer protein at atomic resolution. They were working with samples of the green sulfur bacterium Prosthecochloris estuarii, specimens that had been collected from the coastal waters of New Zealand, where these ancient organisms flourish in the low-oxygen, low-light environments of tidal sediments. Using the technique of X-ray crystallography, Fenner and Matthews were able to map the positions of the atoms within this protein complex, revealing its architecture in exquisite detail, and what they found was a structure of elegant simplicity, a protein scaffold that held within it 7 molecules of bacteriochlorophyll, arranged in a precise geometric pattern that seemed almost deliberately designed to facilitate the movement of energy. The structure that Fenner and Matthews revealed was remarkable in several respects, not least because it was one of the first membrane protein structures ever solved to high resolution, a technical achievement of considerable magnitude at the time. The protein formed a trimer, 3 identical subunits arranged in a 3-fold symmetric pattern, with each subunit cradling its 7 bacteriochlorophyll molecules in a distinctive spatial arrangement. The distances between these chlorophyll molecules, and their relative orientations, seemed to suggest that they were positioned precisely to allow energy to flow between them, almost like stepping stones across a stream, and this observation planted the seed of a question that would take decades to fully answer, which was, by what mechanism does energy actually move through this exquisite molecular architecture? And why is the arrangement so precise if ordinary classical processes would suffice? In recognition of the contributions of all 3 scientists, the protein came to be known as the Fenner-Matthews-Olson complex, or simply the FMO complex, and it became a model system for studying the physics of photosynthetic energy transfer, precisely because of its relative simplicity and the clarity with which its structure was known. Over the following decades, researchers returned to this complex again and again, probing it with increasingly sophisticated experimental techniques, seeking to understand how it accomplished its task with such remarkable efficiency, for it had long been known that photosynthesis, taken as a whole, achieves something quite extraordinary in energetic terms. When a photon of light is absorbed by the antenna systems of a photosynthetic organism, the energy of that photon must travel through a series of molecular components before it reaches the reaction center, and this journey, while it spans only nanometers in distance, passes through multiple transfers between different molecules. In any energy transfer process, there is typically some loss, some dissipation of useful energy into heat or other unusable forms, and yet photosynthesis manages to deliver more than 95% of the absorbed light energy to the reaction center, a figure that seems almost impossibly high when compared to the efficiencies achieved by human-engineered systems. The best commercial solar panels available today, for instance, typically convert only about 20% of incident light energy into electrical energy, with much of the remainder being lost to heat and other inefficiencies, and even the most advanced laboratory photovoltaics struggle to exceed 40% efficiency under concentrated sunlight. That plants, and algae, and ancient bacteria should so dramatically outperform our best engineering efforts suggested that nature had discovered something about energy transfer that we had not yet understood, some trick or principle that allowed these biological systems to achieve near-perfect efficiency in conditions that would seem impossibly noisy and disordered to a physicist designing an idealized energy transfer system. This brings us to the spring of 2007, and specifically to the 12th of April of that year, when the journal Nature published a paper that would fundamentally alter how scientists thought about quantum mechanics and living systems. The lead author of this paper was Greg Engel, and the work had been conducted at the Lawrence Berkeley National Laboratory under the direction of Graham Fleming, using a team of researchers who had developed an extraordinarily precise technique for probing the dynamics of energy transfer at timescales almost inconceivably short to everyday human experience. The technique they employed was called two-dimensional femtosecond spectroscopy, and to appreciate what this involves, we must first understand something about femtoseconds themselves. A femtosecond is 1/millionth of 1 billionth of a second, a duration so brief that light itself, traveling at 300,000 kilometers per second, covers only about 300 nanometers in that time, roughly the wavelength of ultraviolet light. To probe events at the femtosecond timescale is to watch the actual motions of energy as it moves between molecules, to catch the system in the act of transferring excitation from one place to another, and this requires laser pulses of extraordinary brevity and precision. The two-dimensional aspect of the spectroscopy refers to the way in which sequences of these ultra-short pulses are used to map out the correlations between different energy states, creating a kind of fingerprint that reveals not just where the energy is, but where it came from and how it got there. What Engel and the Berkeley team observed, when they applied this technique to the FMO complex, was something that many physicists had assumed impossible in a biological system at anything approaching normal temperatures. They found evidence of quantum coherence, a phenomenon in which the energy exists not in one discrete location or another, but in a superposition of multiple states simultaneously, with the different possibilities interfering with one another in the manner characteristic of quantum mechanical waves. This coherence manifested itself as a rhythmic oscillation in the signals they recorded, a kind of quantum beating that revealed the system exploring multiple energy transfer pathways at once, rather than committing to a single classical path through the molecular landscape. The skepticism from the physics community was immediate and, in many ways, entirely reasonable. Quantum coherence is a famously fragile phenomenon, one that typically requires systems to be isolated from their environments and cooled to temperatures approaching absolute zero, where the chaotic thermal motions of surrounding molecules cannot disrupt the delicate phase relationships that define quantum superposition. The interior of a living cell, by contrast, is a warm, wet, crowded environment, filled with water molecules jostling about and proteins flexing and vibrating in constant thermal motion. That quantum coherence should survive for any appreciable time in such conditions seemed to contradict the basic physics of decoherence, the process by which quantum superpositions are destroyed through interaction with the environment, and many researchers initially suspected that the observed signals must have some other, more prosaic explanation. Yet the data from the Berkeley experiments were difficult to dismiss. The quantum beating signals persisted for approximately 660 femtoseconds at temperatures relevant to biological function, a duration that, while brief in absolute terms, was far longer than theoretical calculations had predicted quantum coherence could survive in such a noisy environment. 660 femtoseconds is long enough for the energy to travel from the antenna to the reaction center, long enough for the coherence to play a meaningful role in the energy transfer process, and this observation suggested that the FMO complex, and perhaps photosynthetic systems more broadly, had somehow found a way to protect quantum coherence from the decoherence that should have destroyed it almost instantly. The implications of this discovery were profound and continue to reverberate through multiple fields of science. If nature had indeed found a way to harness quantum effects for energy transfer in the warm, wet conditions of living cells, then perhaps our assumptions about the boundary between quantum and classical physics needed revision. Perhaps the biological world was not, as had generally been assumed, too noisy and hot for quantum mechanics to matter, but had instead evolved to exploit quantum phenomena in ways we were only beginning to appreciate. This possibility opened new avenues of research, new questions about whether other biological processes might also depend on quantum effects, and new opportunities for learning from nature's example in the design of artificial systems for energy capture and conversion. The story of how this discovery emerged, From Olson's patient work in the 1960s through the structural revelations of Fenner and Matthews in the 1970s to the spectroscopic breakthroughs of the Berkeley team in 2007, illustrates something important about how science progresses. Each contribution built upon those that came before, each answered some questions while raising new ones, and the full significance of the earlier work only became apparent in light of what followed. The green sulfur bacteria that Olson studied, the New Zealand specimens that yielded their secrets to Fenner and Matthews, the FMO complex that became a model system for decades of subsequent research, all of these elements came together in a discovery that no single researcher could have anticipated, a discovery that emerged from the collective, cumulative effort of a scientific community working across generations. As we consider this story, and as perhaps the details begin to blur pleasantly into the general impression of careful work and surprising findings, it is worth holding on to the central wonder of what was discovered. Plants and bacteria, organisms that seem so ordinary and familiar, may be doing something quantum mechanical as they go about the mundane business of capturing sunlight, and they may be doing it better than our best theories predicted possible. The efficiency with which they harvest light, that remarkable 95% and more, may depend on principles that operate at the very foundations of physical reality, in the strange realm where particles are also waves and where systems can explore multiple possibilities at once before settling into a definite outcome. And this realization, far from diminishing the quiet miracle of a leaf turning sunlight into sugar, only deepens our appreciation for the extraordinary sophistication that evolution has achieved, working with nothing more than random variation and natural selection across the vast expanses of deep time. And so we come now to the chlorophyll molecule itself, that remarkable pigment which gives leaves their characteristic color and which sits at the very heart of all photosynthetic life on Earth. If you were to examine a single molecule of chlorophyll under conditions that allowed you to see its atomic structure, you would find something rather elegant in its complexity, a flat ring system surrounding a single atom of magnesium, with a long hydrocarbon tail extending outward like the anchor of a kite. The molecular formula, for those who find comfort in such precise notation, is C55H72 mg/N4O5, which is to say that each molecule contains 55 carbon atoms, 72 hydrogen atoms, a single magnesium atom at its center, 4 nitrogen atoms arranged in a ring around that magnesium, and 5 oxygen atoms distributed at various positions throughout the structure. This may seem like a great many atoms to keep track of, and indeed it is. But nature assembles these molecules with remarkable consistency in every green plant and photosynthetic bacterium across the entire planet. The story of how we came to understand this molecular structure is itself a gentle narrative of patient scientific work, and it takes us back more than a century to the laboratories of Richard Willstatter, a German chemist whose meticulous investigations into plant pigments would earn him the Nobel Prize in Chemistry in 1915. Willstatter had spent years carefully isolating chlorophyll from plant material, separating it into its component forms, and subjecting these purified samples to the analytical techniques available at that time. Through systematic degradation reactions and careful elemental analysis, he was able to determine the empirical formula of chlorophyll and to establish that the magnesium atom sat at the very center of the molecule, coordinated by 4 nitrogen atoms in what we now recognize as a porphyrin ring system. This was painstaking work, requiring the processing of enormous quantities of plant material to obtain enough pure chlorophyll for analysis, and Willstatter pursued it with the sort of quiet dedication that characterizes so much of important scientific progress. His Nobel lecture, delivered in Stockholm, described this work in the measured tones of someone who understood that scientific truth emerges slowly, through the accumulation of careful observations rather than through dramatic revelations. The magnesium center of chlorophyll is not merely a structural curiosity but plays an essential role in the molecule's ability to interact with light. This metal ion, held in place by coordination bonds to the 4 nitrogen atoms of the porphyrin ring, influences the distribution of electrons throughout the conjugated ring system and thereby determines which wavelengths of light the molecule will absorb and which it will allow to pass through or reflect away. When we measure the absorption spectrum of chlorophyll dissolved in a suitable solvent, we find 2 prominent peaks, one in the blue region of the visible spectrum at approximately 430 nanometers, and another in the red region at approximately 662 nanometers. These specific wavelengths correspond to photons whose energy matches the energy required to promote electrons within the chlorophyll molecule from their ground state to various excited states, and it is this matching of photon energy to molecular energy levels that underlies all of photosynthetic light harvesting. The wavelengths between these 2 absorption peaks, particularly those in the green and yellow portions of the spectrum spanning roughly 500 to 565 nanometers, are not efficiently absorbed by chlorophyll and are instead reflected or transmitted. This is, of course, why plants appear green to our eyes, for the light that reaches us after interacting with a leaf is enriched in precisely those green wavelengths that the chlorophyll molecules have declined to absorb. There is something rather peaceful in this explanation, the idea that the color of forests and meadows and houseplants on windowsills is simply the visible signature of quantum mechanical selection rules playing out at the molecular level, the leftover light from an ongoing process of energy harvesting that has been occurring continuously for billions of years. Within the photosynthetic membranes of plant cells, chlorophyll molecules do not operate in isolation but are instead organized into elaborate structures called antenna complexes, and it is here that the quantum mechanical behavior of light harvesting becomes particularly interesting. A typical antenna complex contains between 200 and 300 individual chlorophyll molecules, along with various accessory pigments such as carotenoids, all held in precise spatial arrangements by a scaffold of protein molecules. The geometry of this arrangement is not random but has been refined by billions of years of evolutionary selection to optimize the capture of light energy and its transfer toward the reaction centers where the actual chemistry of photosynthesis occurs. Each chlorophyll molecule is positioned at a specific distance and orientation relative to its neighbors, and these precise geometrical relationships determine how energy flows through the antenna after a photon is absorbed. The most abundant of these antenna structures is known as light harvesting complex II, often abbreviated as LHCII, and this protein pigment assembly holds the distinction of being the most abundant membrane protein on Earth. This is worth pausing to consider. The idea that of all the proteins embedded in all the biological membranes across our entire planet, from bacterial cell membranes to the organelles of plants and algae, this one particular light harvesting complex is more numerous than any other. Each complex contains 14 chlorophyll molecules and 4 carotenoid molecules arranged around a core of 3 protein subunits, and these complexes are packed at high density into the thylakoid membranes of chloroplasts, creating an extensive network for capturing photons across the visible spectrum. The sheer abundance of LHCII reflects the fundamental importance of light harvesting to life on Earth and the evolutionary pressure to maximize the efficiency of this process. When a photon of appropriate energy strikes one of the chlorophyll molecules within an antenna complex, something quite remarkable happens on an almost unimaginably short timescale. within femtoseconds of photon absorption, which is to say within millionths of a billionth of a second, the energy of the photon is converted into a peculiar quantum mechanical entity known as an exciton. An exciton is best understood as a bound pair consisting of an excited electron and the positively charged hole it leaves behind in the molecule's electron distribution, and this pairing arises from the electrostatic attraction between the negative electron and the positive hole. The exciton is not localized on a single chlorophyll molecule but can spread across multiple pigments simultaneously, existing in what physicists call a delocalized state that spans several molecules at once. This delocalization is a distinctly quantum mechanical phenomenon, impossible to explain using classical physics, and it plays an important role in the efficiency of energy transfer through the antenna complex. The behavior of these excitons and the pathways by which energy moves through antenna complexes were illuminated by the careful experimental work of Robert Emerson, a plant physiologist at the University of Illinois whose investigations in the middle decades of the 20th century revealed unexpected features of photosynthetic light harvesting. In 1943, Emerson and his colleague Charlton Lewis published observations of what became known as the red drop effect, a phenomenon in which the quantum yield of photosynthesis dropped sharply when plants were illuminated with light of wavelengths longer than about 680 nanometers. This was puzzling because chlorophyll can still absorb photons at these longer red wavelengths, and one might have expected photosynthesis to continue efficiently as long as absorption occurred. The red drop suggested that something more complex was happening, that the photosynthetic apparatus had requirements beyond simply capturing photons. Emerson continued to investigate this puzzle over the following years, and in 1957 he reported an even more surprising discovery known as the enhancement effect, or sometimes the Emerson enhancement effect. When plants were illuminated simultaneously with far-red light beyond 680 nanometers and with shorter wavelength red light around 650 nanometers, the rate of photosynthesis was significantly greater than the sum of the rates observed when each wavelength was provided alone. This synergistic effect implied that photosynthesis involves not one but two distinct light-dependent processes, each with somewhat different wavelength requirements, and that both must operate together for maximal efficiency. This insight, derived from patient measurement of oxygen evolution rates under different illumination conditions, laid essential groundwork for our modern understanding of the two photosystems that operate in series within the thylakoid membrane. Emerson did not live to see the full development of this model, passing away in 1959, but his careful experiments provided crucial clues that guided subsequent researchers toward a comprehensive picture of photosynthetic electron transport. The quantum dance of chlorophyll, if we may describe it in such terms, involves the coordinated absorption of photons across hundreds of pigment molecules, the rapid formation and migration of excitons through precisely arranged antenna complexes, and the eventual delivery of this captured energy to reaction centers where it drives the chemistry of water splitting and carbon fixation. All of this occurs with remarkable efficiency, with some antenna systems delivering more than 90% of absorbed photon energy to their reaction centers despite the numerous transfer steps involved. This efficiency arises in part from the quantum mechanical nature of the energy transfer process, in which excitons can sample multiple pathways simultaneously and thereby find optimal routes through the antenna network. The protein scaffolds that hold chlorophyll molecules in place within antenna complexes are not passive structural elements but active participants in tuning the optical and electronic properties of the bound pigments. The local environment around each chlorophyll, including the precise positioning of nearby amino acid residues and the presence of bound water molecules, shifts the absorption spectrum of that particular pigment by small but significant amounts. These shifts create an energy landscape within the antenna, with some chlorophylls absorbing at slightly higher energies and others at slightly lower energies, and this landscape guides the flow of excitons downhill toward the reaction center. It is rather like a gentle funnel, with energy entering at various points around the rim and flowing naturally toward the center where it can be put to use. The timescales involved in this process span an enormous range, from the femtosecond formation of excitons through the picosecond migration of energy across the antenna to the nanosecond and longer timescales of the chemical reactions that ultimately fix carbon and release oxygen. At each stage, the physical processes are well matched to the requirements of the next stage, with faster processes feeding into slower ones in a cascade that maintains high efficiency throughout. The entire system represents a remarkable solution to the engineering problem of capturing dilute photon streams and concentrating their energy for chemical work, a solution that has been refined through countless generations of evolutionary optimization and that continues to inspire human engineers seeking to develop artificial photosynthetic systems. As we settle deeper into consideration of these molecular antennae, it is worth remembering that every green surface we encounter in daily life, every lawn and forest and algal bloom, contains astronomical numbers of these chlorophyll molecules carrying out their quantum dances in parallel. The familiar green of the natural world is the visible evidence of a process that operates at the boundary between quantum physics and biology, where individual photons interact with individual molecules according to the strange rules of quantum mechanics and where the resulting energy transformations sustain nearly all life on Earth. This is not a dramatic observation but rather a quietly remarkable one, the sort of thing that might accompany you gently into sleep, the knowledge that the physics of the very small underpins the biology of the everyday in ways that patient scientists like Willstatter and Emerson spent their careers carefully uncovering. For a very long time, one of the most fundamental assumptions in physics was that quantum mechanical effects, the strange and delicate behaviors that govern the smallest scales of reality, could only be observed under the most extreme and carefully controlled conditions. This assumption seemed entirely reasonable, and it was supported by decades of experimental evidence gathered in laboratories around the world. The thinking went something like this: quantum effects such as superposition, where a particle exists in multiple states simultaneously, and coherence, where quantum waves maintain their phase relationships over time, are extraordinarily fragile phenomena. They are disrupted by the slightest interaction with the surrounding environment, a process known as decoherence, and this disruption happens faster when there is more thermal energy present, which is to say, when things are warmer. Therefore, if you wished to observe quantum behavior in any meaningful way, you needed to cool your experimental apparatus to temperatures approaching absolute zero, that theoretical point at which all thermal motion ceases entirely, roughly minus 273 degrees Celsius. At such temperatures, the random jiggling of atoms is reduced to almost nothing, and the delicate quantum states have a chance to persist long enough to be measured. This is why quantum computing research, for instance, requires elaborate cooling systems that bring processes down to temperatures colder than outer space, and why so much of fundamental physics takes place in cryogenic environments where liquid helium provides the necessary thermal stillness. Given all of this well-established understanding, the idea that quantum coherence might play a functional role in biological systems operating at body temperature seemed, to put it gently, rather unlikely. Living things are warm. They are wet. They are filled with constantly moving molecules and ions, a ceaseless thermal chaos that should destroy any quantum coherence almost instantaneously. A leaf on a tree sits in sunlight at perhaps 25 degrees Celsius, which is nearly 300 degrees above absolute zero on the Kelvin scale, and its internal environment is a soup of water molecules, proteins, lipids, and metabolites all bumping into one another with considerable thermal energy. The conventional wisdom suggested that any quantum effects in such an environment would be washed out within femtoseconds, far too quickly to be relevant to biological function. This view was so widely accepted that most biologists gave no thought to quantum mechanics when considering how photosynthesis worked, and most physicists saw no reason to look for coherent quantum phenomena in warm, messy biological systems. The two fields existed largely in separate intellectual spaces, each assuming the other had little to contribute to their particular problems. This comfortable separation began to dissolve in the early 2000s, when a research group at the University of California, Berkeley, led by Graham Fleming, developed experimental techniques sophisticated enough to peer into the extraordinarily rapid dynamics of photosynthetic energy transfer. Fleming, a physical chemist with expertise in ultrafast spectroscopy, had spent years refining methods for observing molecular processes that unfold on timescales of femtoseconds and picoseconds, durations so brief that light itself travels only a fraction of a millimeter. His laboratory became home to an elegant and powerful technique known as two-dimensional electronic spectroscopy, which built upon earlier spectroscopic methods but added a crucial dimension of information. In conventional spectroscopy, you might shine light on a sample and measure what wavelengths are absorbed or emitted, giving you a kind of one-dimensional signature of the molecular species present. Two-dimensional electronic spectroscopy goes further by using precisely timed sequences of ultra-short laser pulses, each lasting only tens of femtoseconds, and then analyzing how the sample responds to these pulses as a function of both the excitation frequency and the detection frequency, as well as the time delays between pulses. The resulting data can be displayed as a two-dimensional map showing correlations between different electronic states, and by watching how these maps evolve over time, researchers can track the flow of energy through a molecular system with unprecedented detail. What Fleming and his colleagues observed when they applied this technique to photosynthetic complexes was not what anyone had expected. In experiments on the Fenner-Matthews-Olson complex, that small protein from green sulfur bacteria we discussed earlier, they found evidence of quantum coherence persisting for timescales that seemed impossibly long given the temperature of the sample. The experimental signatures they observed showed beating patterns, oscillations in the signal that indicated quantum superposition states were being maintained as energy moved through the system. An exciton, that quantum of electronic excitation created when a chlorophyll molecule absorbs a photon, was not simply hopping from one pigment molecule to the next in a classical random walk. Instead, it appeared to be existing in a superposition across multiple chlorophyll molecules simultaneously, spreading out like a wave and exploring many possible pathways at once. The coherent oscillations in their data suggested that these superposition states were lasting for hundreds of femtoseconds, which may not sound like very long in everyday terms, but is actually remarkably extended for quantum coherence in a warm, wet environment. Classical theories of energy transfer, based on the work of Theodore Foster from the mid-20th century, predicted incoherent hopping between pigments, with the exciton making essentially random jumps from one molecule to an adjacent one, like a person stumbling through a dark room by bumping into furniture. The quantum picture suggested something quite different, a wave-like exploration of the energy landscape, allowing the exciton to sample multiple routes simultaneously and thereby find efficient paths to the reaction center. These initial findings from Fleming's laboratory generated considerable excitement but also considerable skepticism, because the experiments were conducted at cryogenic temperatures, around 77 Kelvin, which is still very cold indeed, roughly minus 196 degrees Celsius. Critics quite reasonably pointed out that while these results were interesting, they did not necessarily tell us anything about what happens in living plants at physiological temperatures. Perhaps quantum coherence could survive briefly at cryogenic temperatures but would be completely destroyed in the warmth of a sunny afternoon. The crucial question was whether these effects persisted under biologically relevant conditions, in the warm, wet, noisy environment of an actual photosynthetic organism going about its ordinary business of converting sunlight into chemical energy. This question was addressed directly in a landmark paper published in the journal Nature Physics in 2010, authored by Gregory Scholes and his collaborators, including researchers from the University of Toronto and other institutions. Scholes and his team studied light harvesting complexes from marine cryptophyte algae, single-celled organisms that drift in ocean waters and capture sunlight filtering down through the waves above them. Using two-dimensional electronic spectroscopy similar to that developed in Fleming's laboratory, they examined these algal proteins at room temperature, at the ordinary warmth of a living cell, and they found clear evidence that quantum coherence was still present. The Scholes paper was remarkable because it demonstrated that the warm, wet, and noisy environment of a living cell did not, as everyone had assumed, instantly destroy quantum coherence in these photosynthetic systems. The coherent oscillations persisted for timescales comparable to those observed at cryogenic temperatures, lasting for several hundred femtoseconds even in the presence of all the thermal motion and molecular collisions that characterize biological conditions. This was the impossible made real, the demonstration that quantum mechanical effects could survive and potentially function in the least quantum-friendly environment imaginable. The finding prompted a fundamental reconsideration of how we think about the relationship between quantum physics and biology, and it launched an entirely new field of research that came to be known as quantum biology. Understanding what quantum coherence actually means in this context requires a bit of careful explanation, because the term coherence is used in several different ways in physics, and its specific meaning here is quite particular. When we say that an exciton exhibits quantum coherence across multiple chlorophyll molecules, we mean that the excitation is not localized on any single pigment but exists in a superposition of being on several pigments simultaneously. You might imagine it, very loosely, as the electronic excitation being smeared out across a group of chlorophyll molecules, with definite phase relationships between the different components of this superposition. These phase relationships are what makes the state coherent rather than simply mixed or averaged. A classical mixed state would be like flipping a coin and not looking at the result. The coin is definitely either heads or tails, you just do not know which. A quantum superposition is fundamentally different. The system genuinely is in both states at once, with the different possibilities interfering with one another like overlapping waves. When this coherent superposition evolves over time, the different components can interfere constructively along some pathways and destructively along others, potentially guiding the energy toward particular destinations more efficiently than a random classical walk would allow. The problem that everyone had to grapple with, once these experimental findings became established, was explaining how coherence could possibly survive in such an unfavorable environment. This became known informally as the warm, wet, and noisy problem, because those three words neatly summarize everything that should destroy quantum coherence. Warmth means thermal energy, which kicks molecules around randomly and disrupts delicate quantum states. Wetness means water, that universal solvent whose molecules are in constant motion and constantly interacting with everything around them through hydrogen bonding and other forces. Noise means the general fluctuating environment of a cell, the endless molecular collisions and vibrations that constitute the thermal bath surrounding any biological structure. By all rights, this environment should cause decoherence, the loss of those precious phase relationships, within femtoseconds at most. And yet the coherence was lasting for hundreds of femtoseconds, long enough to be potentially relevant for guiding energy transfer. One of the most intriguing theoretical proposals for resolving this puzzle came from Alexandra Olaya-Castro and her colleagues at University College London. Working with sophisticated theoretical models of how quantum systems interact with their environments, Olaya-Castro proposed something that initially seemed counterintuitive. Perhaps the protein environment was not merely a source of noise that coherence had to survive despite, but was actually playing an active role in protecting and even enhancing coherence. Her theoretical work suggested that the vibrations of the protein scaffold surrounding the chlorophyll pigments, rather than being purely destructive, could couple to the electronic excitations in ways that sheltered the coherent dynamics from other sources of decoherence. The protein might be acting as a kind of protective cradle, its own internal motions tuned through evolution to support rather than destroy the delicate quantum states passing through it. This idea represented a profound shift in perspective, from viewing the biological environment as an obstacle that quantum effects must somehow overcome, to viewing it as an essential partner that might enable quantum behavior in ways that would be impossible in a simpler, more isolated system. The timescales involved in all of this are worth dwelling on for a moment, because they give a sense of just how rapidly these processes unfold. A femtosecond is 1/4,000,000th of a second, or 1/1,000,000th of a billionth of a second, a duration almost impossible to grasp in intuitive terms. If a femtosecond were scaled up to 1 second, then 1 second would correspond to roughly 32 million years. Light travels about 300 nanometers in a femtosecond, roughly the diameter of a small virus. When we say that quantum coherence persists for hundreds of femtoseconds in these photosynthetic complexes, we are talking about durations on the order of perhaps 200 to 500 femtoseconds, which is still extraordinarily brief by any everyday standard but is surprisingly long by the standards of quantum dynamics in warm environments. This timescale is comparable to the time required for energy to transfer between neighboring chlorophyll molecules, meaning that the coherence lasts long enough to potentially influence the outcome of these transfer events. Whether this influence actually improves the efficiency of photosynthesis, or whether it is simply an interesting side effect with no functional significance, remains a topic of ongoing research and debate, but the mere fact that coherence exists at these timescales was enough to overturn decades of assumptions about the incompatibility of quantum mechanics and warm biological systems. The wave-like character of quantum energy transfer stands in contrast to the classical hopping picture that had dominated thinking about photosynthesis for half a century. In the classical Foster model, energy transfer between pigments is essentially a series of random jumps, with the exciton hopping from one molecule to a neighboring one based on the overlap of their electronic states and the thermal fluctuations of the environment. This process is inherently stochastic and diffusive, like a drop of ink spreading through water. The quantum coherent picture suggests something more directed, with the wave-like exciton sampling multiple pathways simultaneously and potentially finding optimal routes to the reaction center through constructive interference. Whether photosynthetic organisms have evolved to exploit this quantum speedup, or whether the observed coherence is simply a byproduct of the molecular physics involved with no adaptive significance, is a question that researchers continue to investigate with ever more sophisticated experiments and theories. If you were to travel to the great thermal basins of Yellowstone National Park, to those places where the Earth breathes out its ancient heat through pools of water that shimmer and steam beneath the open sky, you would find, along the edges of certain hot springs, delicate films of color that seem almost painted upon the rocks and sediments. These colors range from deep purples to rich magentas, and they belong to some of the oldest living organisms on our planet, organisms that have been quietly harvesting the energy of sunlight for longer than almost anything else that breathes or grows upon this earth. These are the purple bacteria, and their story stretches back through an almost incomprehensible span of time, some 3.5 billion years, to an era when our planet was young and strange and the very air was unlike anything we would recognize today. The purple bacteria, including species with names like Rhodopseudomonas and Rhodobacter, thrive in environments that might seem inhospitable to us, in hot springs and stagnant ponds and the murky depths of lakes where oxygen is scarce or entirely absent. They are found not only in the famous geothermal features of Yellowstone but also in similar extreme environments scattered across the globe, from the volcanic regions of Iceland to the thermal pools of New Zealand, and in countless quieter places where warm, still water meets the fading light of the sun. These organisms are part of a group known as the anoxygenic phototrophs, a term that simply means they are capable of using light for energy without producing oxygen as a byproduct, and this detail, seemingly small, is in fact a window into the deep history of life itself. To understand why these purple bacteria are so significant, one must imagine the Earth as it was billions of years ago, a world in which the atmosphere contained almost no free oxygen, in which the seas and skies were filled instead with gases like carbon dioxide and methane and hydrogen sulfide. In this ancient world, the earliest forms of photosynthesis did not split water molecules to release oxygen, as the photosynthesis of plants and cyanobacteria does today, but instead drew upon other chemical sources, compounds like hydrogen sulfide or organic acids, to provide the electrons needed for the light-driven chemistry of life. The purple bacteria are living descendants of these ancient photosynthesizers, and in their simple, elegant biochemistry, we can glimpse the mechanisms that first allowed life to capture and store the energy of the sun. The recognition that bacteria could photosynthesize at all was itself a discovery that took many years to establish and accept. In the 1930s, a Dutch-American microbiologist named Cornelis van Niel, working at the Hopkins Marine Station of Stanford University in Pacific Grove, California, undertook a careful and methodical study of these purple and green bacteria, seeking to understand how they obtained their energy and how their metabolism compared with that of plants. Van Niel was a meticulous scientist, patient and thorough, and through a series of elegant experiments and comparative analyses, he demonstrated that these bacteria were indeed photosynthetic, that they used light energy to drive the synthesis of organic compounds, but that they did so without releasing oxygen into their surroundings. His work laid the foundation for our modern understanding of photosynthesis as a process with many variations, a process that evolved in different forms long before the oxygen-producing photosynthesis of cyanobacteria and plants transformed the atmosphere of our planet. Van Niel proposed a general equation for photosynthesis that could accommodate both the oxygen-producing kind and the anoxygenic kind performed by purple bacteria, and in doing so, he unified our understanding of these processes and revealed the deep chemical logic that underlies them all. He recognized that photosynthesis, at its heart, is a process of electron transfer, of using the energy of light to move electrons from a donor substance to an acceptor, and that the specific identity of that donor, whether water or hydrogen sulfide or something else entirely, determines whether oxygen is released. In the case of purple bacteria, the electron donor is not water but rather compounds like hydrogen sulfide or organic molecules, and so no oxygen escapes into the environment, which is why these organisms could thrive in the oxygen-free world of early Earth. For many decades after Van Niel's pioneering work, scientists continued to study the purple bacteria and their photosynthetic machinery, seeking to understand the precise molecular structures that enabled these organisms to capture light and convert its energy into chemical form. This was no simple task, for the proteins responsible for photosynthesis are embedded within the membranes of the bacterial cells, and membrane proteins are notoriously difficult to isolate, purify, and analyze. They are large and complex molecules, surrounded by a delicate shell of lipid molecules, and they tend to lose their structure and function when removed from their native environment. For years, the detailed architecture of the photosynthetic reaction center, the molecular machine at the heart of bacterial photosynthesis, remained frustratingly out of reach. The breakthrough came in the early 1980s, at the Max Planck Institute of Biochemistry in Martinsried, a small town just south of Munich in Germany. There, a team of 3 scientists, Johann Deisenhofer, Robert Huber, and Hartmut Michel, embarked on a project that would change our understanding of biological membranes and earn them the Nobel Prize in Chemistry. Hartmut Michel, in particular, had been working for several years on the challenge of crystallizing membrane proteins, a task that many considered impossible, since proteins typically need to be coaxed into forming orderly crystals in order for their structures to be determined by X-ray diffraction, and membrane proteins, with their awkward coats of lipid, seemed unlikely candidates for such treatment. Michel, however, persevered with quiet determination, testing countless combinations of conditions and detergents and precipitants, and in 1982, he succeeded in producing crystals of the photosynthetic reaction center from a purple bacterium called Rhodopseudomonas viridis, a species closely related to the organisms found in hot springs and other extreme environments. These crystals were small and fragile, but they were sufficient for the task at hand, and with the help of Deisenhofer, who specialized in the mathematical analysis of X-ray diffraction patterns, and Huber, who oversaw the technical aspects of the work, the team set about determining the 3-dimensional structure of this remarkable protein complex. The work was painstaking and slow, requiring the collection of vast amounts of data and the careful interpretation of patterns that revealed, piece by piece, the arrangement of atoms within the reaction center. By 1984, the team had produced the first detailed structure of a membrane protein, a milestone in the history of biology and chemistry, and in 1985, they published their findings, revealing the molecular architecture of nature's first solar panels. The significance of this achievement cannot be overstated, for it opened the door to the study of countless other membrane proteins, including the ion channels and receptors and transporters that govern so much of cellular life, and it provided a template for understanding how biological membranes carry out their essential functions. The structure that Deisenhofer, Huber, and Michel revealed was both beautiful and instructive, a precise arrangement of protein and pigment that seemed perfectly designed for its task of capturing light and directing the flow of electrons. At the heart of the reaction center are two protein subunits, known simply as L and M, which together form a scaffold that holds in place the pigment molecules responsible for absorbing light and initiating the transfer of electrons. These pigment molecules include 4 bacteriochlorophylls and 2 bacteriophytons, closely related to the chlorophyll found in plants but adapted to absorb light of slightly different wavelengths, which is why the bacteria appear purple rather than green. The bacteriochlorophylls are arranged in a specific pattern, with 2 of them positioned very close together at the center of the reaction center, forming what is called the special pair, and it is here that the magic of photosynthesis begins. When a photon of light strikes the special pair, its energy is absorbed and one of the electrons in these pigments is boosted to a higher energy state, a state from which it can escape and begin its journey through a chain of electron acceptors embedded within the protein. The first step in this journey is astonishingly fast, occurring in approximately 3 picoseconds, which is 3/trillionths of a second, a span of time so brief that light itself travels only about a millimeter in that interval. This initial electron transfer, from the special pair to a nearby bacteriophyton molecule, sets off a cascade of subsequent transfers, each step moving the electron farther from its origin and storing its energy in a more stable form. The protein structure ensures that these transfers proceed in an orderly and efficient manner, with each pigment and cofactor positioned precisely to receive the electron and pass it along, like a series of hands in a relay race, each one perfectly placed to catch and throw the baton. The final destination of the electron is a quin-1 molecule, which accepts 2 electrons and 2 protons to become a reduced quin-1, and this molecule then diffuses through the membrane to participate in further biochemical reactions. The elegance of this arrangement, the precise positioning of each component, the exquisite timing of each transfer, speaks to billions of years of evolutionary refinement, a slow and patient optimization that has produced a molecular machine of remarkable efficiency. The reaction center of purple bacteria captures and converts light energy with a quantum efficiency that approaches 100%, meaning that nearly every absorbed photon leads to a successful electron transfer, and this efficiency is a consequence of the quantum mechanical properties of the system, properties that we will explore in later sections of this narration. In recognition of their groundbreaking work, Johann Deisenhofer, Robert Huber, and Hartmut Michel were awarded the Nobel Prize in Chemistry in 1988, an honor that acknowledged not only the specific achievement of determining the reaction center structure but also the broader impact of their methods on the study of membrane proteins. The techniques they developed and refined at the Max Planck Institute have since been applied to hundreds of other proteins, revealing the structures of molecules that control nerve impulses, regulate cellular metabolism, and defend against disease. And in this way, the humble purple bacterium has contributed to advances in medicine and biology far beyond its own quiet existence in the hot springs and ponds of the world. The anoxygenic photosynthesis of these bacteria, though less familiar than the oxygen-producing kind, is no less important to the history of life on Earth. For perhaps 1 billion years or more, before the evolution of cyanobacteria and the great oxygenation event that transformed our planet's atmosphere, the only photosynthesis that existed was of this ancient, oxygen-free variety. The purple bacteria and their relatives were the pioneers, the first organisms to tap into the vast and inexhaustible energy of sunlight, and in doing so, They laid the biochemical foundations upon which all subsequent photosynthesis would be built. When you close your eyes tonight and let your thoughts drift towards sleep, you might imagine those hot springs and their quiet films of purple life, ancient organisms still carrying on the work they began so long ago, capturing photons and shuffling electrons through their delicate protein scaffolds, indifferent to the passage of eons. They ask nothing of us and offer only a gentle lesson in patience and persistence, a reminder that even the most profound transformations of the living world began with small and simple steps, repeated over and over across the vast expanse of time. The purple bacteria are still there, still basking in the warmth and the light, and their slow, steady chemistry continues beneath the open sky, as it has for 3.5 billion years, and will, perhaps, for billions more. And so we arrive now at the reaction center, that quiet and remarkable place within the photosynthetic machinery where the strange behaviors of the quantum world meet the more familiar processes of chemistry, and where light energy is converted, through a series of patient and carefully orchestrated steps, into the chemical energy that will eventually sustain nearly all life on Earth. This is a gentle place to rest our attention for a while, for the reaction center operates with a kind of unhurried precision that mirrors the calm and methodical pace we might wish to adopt ourselves as we settle deeper into relaxation, allowing the soft details of molecular biology to wash over us like a warm and reassuring tide. The reaction center we shall focus upon is found within a larger complex known as Photosystem II, which sits embedded in the thylakoid membranes of chloroplasts, those green-tinted organelles that give leaves their familiar color. Photosystem II is, in many ways, the most extraordinary of the photosynthetic complexes, for it performs a task that no other biological system on Earth can accomplish, and that task is the splitting of water molecules into their constituent parts, releasing oxygen gas as a byproduct and capturing the electrons and protons that will be needed to drive the rest of the photosynthetic process. This water-splitting capability is so chemically demanding, so energetically difficult, that for billions of years before photosynthesis evolved this particular trick, the Earth's atmosphere contained almost no free oxygen at all, and the planet was a very different place indeed. The discovery that Photosystem II operates through a series of discrete steps, rather than in one continuous process, emerged from careful experimental work conducted in Paris during the 1960s, when a French biophysicist named Pierre Joliot began measuring the patterns of oxygen release from illuminated chloroplasts with extraordinary precision. Joliot, working at what would become the Institut de Biologie Physico-Chimique, noticed something curious and beautiful in his data, which was that the oxygen released by photosynthetic membranes did not emerge smoothly and continuously as one might expect, but rather appeared in a characteristic oscillating pattern, with peaks of oxygen production occurring after every fourth flash of light. This four-flash periodicity suggested that the water-splitting machinery was cycling through a series of distinct states, accumulating the necessary oxidizing power one photon at a time, until it had gathered enough energy to split two water molecules and release one molecule of oxygen gas. It was a finding that would shape our understanding of photosynthetic oxygen evolution for decades to come. At the heart of Photosystem II's water-splitting capability lies a small but astonishing cluster of metal atoms known as the manganese-calcium cluster, or more precisely, the Mn4Ca5 cluster, which contains 4 manganese atoms, 1 calcium atom, and 5 oxygen atoms arranged in a specific geometric configuration that has been refined by evolution over billions of years. This tiny cluster, sometimes called the oxygen evolving complex, is the only biological structure known that can oxidize water molecules, stripping away their electrons and protons and leaving behind molecular oxygen as the quiet and essential byproduct that accumulates in our atmosphere and allows us to breathe. The manganese atoms within this cluster cycle through different oxidation states as the reaction proceeds, each absorbing the oxidizing power generated when the reaction center captures a photon, and together they accumulate enough chemical potential to drive the otherwise impossible reaction of water oxidation. The mechanism by which this cluster operates was elegantly described by a Dutch-American biochemist named Bessel-Koch, who in 1970 published a model that explained Joliot's oscillating oxygen patterns in terms of what he called S-states, a series of 5 intermediate states labeled S0 through S4 that represent the progressive oxidation of the manganese cluster. In this model, which we now call the Koch cycle or sometimes the S-state cycle, the cluster begins in a relatively reduced state called S0 and, with each photon absorbed by the reaction center, advances one step along the cycle, from S0 to S1, then from S1 to S2, then from S2 to S3, and finally from S3 to the brief and transient S4 state, which is so unstable and short-lived that it immediately reacts with water and releases molecular oxygen, returning the cluster to the S0 state and beginning the cycle anew. This means that photosystem 2 must absorb 4 photons, one at a time and with considerable patience, to produce just one molecule of the oxygen we breathe, and this fourfold requirement reflects the fundamental chemistry of water, which contains 4 hydrogen atoms distributed across 2 water molecules, and whose oxidation requires the removal of 4 electrons and 4 protons. To understand how the energy of those captured photons drives this cycle, we must consider the remarkable oxidizing power of the reaction center itself, and here we encounter one of the most extreme examples of biological chemistry known to science. The primary electron donor within photosystem 2 is a special pair of chlorophyll molecules known as P680, so named because they absorb light most strongly at a wavelength of 680 nanometers, and when P680 is oxidized, that is, when it loses an electron, it becomes the most powerfully oxidizing species found anywhere in the biological world. The midpoint potential of oxidized P680, written as P680+, is approximately positive 1.2 volts, which represents an extraordinary oxidizing strength, strong enough to pull electrons away from water molecules themselves, something that very few chemical systems, biological or otherwise, can accomplish. This oxidizing power does not act directly upon the manganese cluster, however, for between P680 and the oxygen-evolving complex lies an intermediary, a modified amino acid that serves as a kind of gentle relay point in the electron transfer process. This intermediary is a tyrosine residue, specifically tyrosine 161 of the D1 protein subunit, and it is commonly referred to as YZ to distinguish it from other tyrosines in the complex. When P680 becomes oxidized, it extracts an electron from YZ, converting this tyrosine into what is called a tyrosine radical, a neutral radical species that retains a positive charge in a delocalized form. This tyrosine radical, sometimes written as YZ, then extracts an electron from the manganese cluster, advancing it one step along the S-state cycle, and the presence of this intermediary helps to protect the delicate reaction center from the extreme oxidizing conditions required for water splitting. The role of YZ as this crucial electron transfer relay was elucidated through detailed spectroscopic work, and much of our understanding of its function came from research conducted by Gerald Babcock at Michigan State University during the 1980s and 1990s, where he used electron paramagnetic resonance and other techniques to characterize the radical states involved in this process. The structural details of photosystem II remained somewhat mysterious for many years, known only through indirect methods and clever inference, until advances in X-ray crystallography allowed researchers to visualize the complex directly at atomic resolution. A particularly significant milestone was reached in 2004, when a research group led by James Barber at Imperial College London, working in collaboration with colleagues including So Iwata, published a crystal structure of photosystem II from the cyanobacterium Thermosynechococcus elongatus at a resolution of 3.5 angstroms, which is 3.5 of a nanometer, a scale at which individual amino acids and cofactors could be clearly distinguished. This structure revealed for the first time the detailed arrangement of the manganese-calcium cluster and its relationship to the surrounding protein environment, showing how the cluster is cradled within a pocket formed by specific amino acid residues of the D1 and CP43 protein subunits, and how water molecules are positioned nearby, ready to be oxidized when the cluster reaches the S4 state. Subsequent work by other groups, including Genren Shin and colleagues in Japan, would eventually achieve even higher resolution, but Barber's 2004 structure represented a transformative moment in our understanding, providing a molecular framework that could explain decades of biochemical and spectroscopic observations. To place photosystem II within the broader context of photosynthetic electron transport, we must now consider an elegant conceptual framework known as the Z-scheme, which was proposed in 1960 by Robert Hill and Faye Bendall working at Cambridge University. The Z-scheme describes how electrons flow through the photosynthetic apparatus, from water through photosystem II, then through a series of intermediate carriers, and finally through a second photosystem called photosystem I, before being used to reduce the electron carrier NADP+ to NADPH. When this scheme is drawn as a diagram with the redox potential on the vertical axis and the sequence of electron carriers on the horizontal axis, it traces a shape that resembles the letter Z, hence the name, with two upward jumps representing the light-driven excitation events in the two photosystems, and downward slopes representing the spontaneous flow of electrons between carriers. Hill and Bendall proposed this scheme based on careful analysis of the redox potentials of various photosynthetic components, and their insight unified what had previously seemed like disconnected observations into a coherent and elegant description of how light energy drives electron flow through the photosynthetic membrane. Within this Z-scheme, photosystem II occupies a position of particular thermodynamic significance, for it is here that electrons begin their journey at the extremely low redox potential of water, and must be lifted up to much higher energy levels by the absorption of light. The P680 reaction center, when excited by absorbing a photon or receiving excitation energy from the antenna system, enters an excited state with sufficient reducing power to donate an electron to the primary acceptor, a molecule of Phi-arphatin, and this initial charge separation sets in motion the chain of electron transfers that will eventually lead to oxygen evolution on one side of the complex and plastoquinone reduction on the other. The electron donated by excited P680 travels through a series of carriers, first to Phi-arphatin, then to a tightly bound plastoquinone molecule called Qa, and finally to a more mobile plastoquinone molecule called Qb, which after accepting 2 electrons and 2 protons becomes fully reduced and diffuses away into the membrane to carry its cargo of electrons and protons to the next complex in the chain, Meanwhile, the oxidized P680 that remains after this electron donation possesses that extraordinary oxidizing power we discussed earlier, strong enough to extract electrons from YZ and ultimately from water itself, and it is this dual nature of the reaction center, acting both as a powerful reductant when excited and a powerful oxidant when oxidized, that allows it to bridge the vast thermodynamic gap between water and the downstream electron carriers. The careful positioning of all these components within the protein scaffold of photosystem II ensures that each electron transfer step occurs with high efficiency and appropriate timing, preventing the wasteful recombination of charges and allowing the precious energy of captured photons to be conserved and put to productive use. As we rest here amid these molecular details, allowing the gentle complexities of the reaction center to settle around us like a soft and reassuring blanket, we might reflect on the quiet patience with which photosystem II performs its essential task, absorbing one photon at a time, advancing one step at a time through the Koch cycle, accumulating the oxidizing power needed to split water only through the diligent repetition of this cycle 4 times over. There is no hurry in this process, no dramatic rush toward completion, only the steady and methodical accumulation of chemical potential, the quiet persistence of quantum events leading inexorably toward the release of molecular oxygen, one bubble at a time, into the atmosphere of our planet. And so the reaction center reveals itself as a place where the abstract strangeness of quantum mechanics meets the familiar reliability of chemistry, where photons from the sun are captured and converted with remarkable efficiency into the chemical currencies of life, and where the splitting of water, that most abundant and essential of molecules, provides the electrons that will flow onward through the photosynthetic machinery, carrying with them the energy that sustains nearly all living things. It is a gentle place, this reaction center, a place of patient accumulation and quiet transformation, and we may let our thoughts rest here for a moment, surrounded by manganese atoms and tyrosine radicals, by chlorophylls and quinones, by the soft and steady pulse of light-driven chemistry that has been running, unhurried and essential, for billions of years. In the quiet interior of every photosynthetic cell, electrons are undertaking journeys that classical physics would tell us are simply impossible, and yet these journeys happen billions of times each second, with a reliability that has sustained life on Earth for billions of years. This is the realm of quantum tunneling, a phenomenon so counterintuitive that even the physicists who first described it found themselves struggling to reconcile its predictions with everyday experience, and yet it is precisely this strange quantum behavior that allows plants and photosynthetic bacteria to move electrons with the speed and efficiency that photosynthesis demands. As you settle into a comfortable position and allow your breathing to slow, we can explore together how electrons take these quantum shortcuts through barriers that should, by all classical reasoning, be completely impenetrable. To understand what tunneling means, we must first consider what happens when a particle encounters a barrier in the classical world, the world of our everyday experience. If you roll a ball toward a hill, and the ball does not have enough energy to climb over that hill, it will simply roll back down, unable to reach the other side. This seems obvious, almost trivially so, and for objects like balls and cars and people, this is indeed how the world works. But electrons are not balls, and at the quantum scale, particles do not behave as tiny versions of the objects we can see and touch. Instead, electrons are described by wave functions, mathematical expressions that spread out through space and assign probabilities to finding the electron in various locations. When an electron's wave function encounters a barrier, something remarkable happens. The wave function does not simply stop at the barrier's edge but instead penetrates into the barrier itself, diminishing in amplitude as it goes, and if the barrier is thin enough, a portion of that wave function emerges on the other side. This means there is a finite probability, sometimes quite substantial, that the electron will simply appear on the far side of the barrier without ever having climbed over it, as if it had tunneled straight through the solid wall. The theoretical framework for understanding how electrons tunnel through the protein medium of photosynthetic complexes was developed over decades, with particularly important contributions coming from Rudolf Marcus, a physical chemist who began his work on electron transfer theory at the Polytechnic Institute of Brooklyn in the early 1950s and continued it at the California Institute of Technology through the 1960s and beyond. Marcus was interested in a deceptively simple question, namely what determines the rate at which an electron will transfer from one molecule to another, and his approach combined classical ideas about molecular reorganization with the quantum mechanical treatment of electron motion. The theory he developed, now universally known as Marcus theory, describes how the energy landscapes of the electron donor and acceptor must align for transfer to occur, and how the surrounding medium, whether it be solvent molecules or the amino acids of a protein, must reorganize to accommodate the changed charge distribution. For this work, which proved foundational to our understanding of countless chemical and biological processes, Marcus received the Nobel Prize in Chemistry in 1992, recognition that came nearly 40 years after his initial theoretical insights, a delay that speaks to how long it sometimes takes for the scientific community to fully appreciate the importance of theoretical contributions. Within the reaction centers of photosynthetic organisms, electrons must travel substantial distances through what is essentially a protein medium, hopping from one cofactor to the next along the electron transfer chain. The distances involved are measured in angstroms, a unit equal to 1/10,000,000,000th of a meter, and electrons routinely tunnel through spans of up to 14 angstroms of intervening protein material. To put this in perspective, a typical chemical bond between two atoms is roughly 1 to 2 angstroms in length, so 14 angstroms represents a journey through multiple atomic diameters of seemingly solid matter. In the bacterial reaction center, the first electron transfer step, from the excited special pair of bacteriochlorophylls to the nearby bacteriophyophytin, occurs in approximately 200 picoseconds, and this transfer happens through an edge-to-edge distance of several angstroms with remarkable efficiency. A picosecond is 1/trillionth of a second, a duration so brief that light itself travels only about 3/10 of a millimeter in that time, and yet this is ample time for quantum tunneling to carry an electron to its destination. The protein medium through which these electrons tunnel is not simply a passive barrier but rather an active participant in the transfer process, and this is where the concept of superexchange becomes important. In superexchange, the electron does not simply tunnel through empty space but instead makes use of the electronic orbitals of the intervening atoms, particularly the amino acids that lie along the tunneling pathway. One might imagine the electron borrowing briefly the electronic states of these bridging molecules, using them as a sort of quantum highway that enhances the probability of reaching the destination. The electron never actually occupies these intermediate states in any classical sense, which would require additional energy and slow the process considerably, but rather the quantum wave function extends through these electronic orbitals in a coherent fashion, effectively shortening the tunneling distance. The specific arrangement of amino acids between donor and acceptor cofactors in photosynthetic proteins has been shaped by evolution to optimize these superexchange pathways, with aromatic amino acids like tryptophan and tyrosine often positioned along the tunneling routes, their extended electron clouds providing efficient conduits for the tunneling process. Harry Gray, also working at the California Institute of Technology, made seminal contributions to our understanding of electron tunneling in proteins through elegant experiments using modified proteins. Gray and his colleagues developed techniques to attach light-absorbing metal complexes to specific locations on protein surfaces, allowing them to trigger electron transfer with pulses of laser light and measure the rates with extraordinary precision. By systematically varying the distance between donor and acceptor and mapping the intervening protein structure, Gray's laboratory established quantitative relationships between tunneling rates and the nature of the protein medium. These experiments demonstrated that electrons tunnel much more readily through well-structured regions of protein, particularly through the beta sheets and alpha helices that form the backbone of protein architecture, than through loosely organized loops or through empty space. The insights from these studies have been invaluable not only for understanding natural systems like photosynthesis but also for designing artificial systems that might someday mimic the efficiency of biological electron transfer. One of the most surprising predictions of Marcus theory concerns what happens when an electron transfer reaction is made more thermodynamically favorable. In ordinary chemical intuition, one would expect that making a reaction more downhill, more exergonic in technical language, would always make it proceed faster, since the driving force pushing the reaction forward would be greater. Marcus' theory predicted something quite different, that beyond a certain point, further increasing the driving force would actually slow the reaction down, a phenomenon known as the inverted region. This counterintuitive prediction was initially met with skepticism, but experiments eventually confirmed its existence, providing striking validation of Marcus's theoretical framework. The inverted region arises because electron transfer requires not only sufficient energy but also proper alignment of the quantum mechanical wave functions of donor and acceptor, and when the energy difference becomes too large, this alignment becomes increasingly difficult to achieve. In photosynthetic reaction centers, the energy levels of successive cofactors appear to be tuned by evolution to keep electron transfers operating near the peak of the Marcus rate curve, avoiding both the normal region where transfer would be limited by insufficient driving force and the inverted region where excess driving force would paradoxically slow things down. At the University of Pennsylvania, Christopher Moser and Leslie Dutton dedicated years of careful research to establishing empirical rules for electron tunneling in biological systems. Their approach was both experimental and analytical, measuring tunneling rates in a wide variety of natural proteins and artificial constructs to identify the underlying principles governing these processes. From this extensive body of work emerged what is sometimes called the Moser-Dutton ruler, a set of quantitative relationships that allow scientists to predict tunneling rates based on distance, driving force, and the reorganization energy of the system. Perhaps most remarkably, Moser and Dutton found that the rate of electron tunneling decreases exponentially with distance, falling by roughly a factor of 10 for every 1.7 angstroms of additional separation when the electron tunnels through typical protein material. This simple relationship, emerging from the complex quantum mechanics of the tunneling process, provides a powerful tool for understanding how photosynthetic proteins are designed and how their performance might be optimized or replicated. The distances between cofactors in photosynthetic reaction centers appear to be precisely optimized based on these tunneling rules. Adjacent cofactors in the electron transfer chain are typically separated by edge-to-edge distances of around 10 to 14 angstroms, close enough for tunneling to occur rapidly but far enough to prevent the wasteful backtransfer of electrons. If the cofactors were placed closer together, electrons might tunnel backward as easily as forward, leading to futile cycling that would waste the energy captured from light. If the cofactors were placed farther apart, tunneling rates would drop precipitously, allowing competing processes like charge recombination to rob the system of its hard-won electronic separation. The positioning observed in natural systems represents a careful balance, maintained over billions of years of evolutionary refinement, that maximizes the probability of productive forward electron transfer while minimizing the probability of energy-wasting back reactions. The protein structure surrounding the electron transfer chain does more than simply position the cofactors at optimal distances. It also creates a medium through which tunneling can proceed efficiently. The amino acids that lie between donor and acceptor are not randomly arranged but rather form organized structures, often including stretches of alpha helix or beta sheet that provide covalently bonded pathways for the tunneling electron. Through these structured regions, the exponential decay of tunneling probability with distance is less severe than it would be through disordered material or through vacuum, effectively extending the reach of the tunneling process. Computational studies have mapped the specific tunneling pathways in various photosynthetic proteins, identifying the key amino acids through which the electron's wave function extends and revealing how mutations in these residues can dramatically alter tunneling rates. These insights are not merely academic curiosities but offer guidance for the design of artificial photosynthetic systems, suggesting how proteins or other materials might be engineered to optimize electron transfer for human applications. As we consider these quantum mechanical processes unfolding in the quiet interior of every leaf and every photosynthetic bacterium, there is something deeply restful in recognizing how these ancient molecular machines have mastered the quantum world in service of life. The electrons that tunnel through protein barriers in your houseplants or in the trees outside your window are following rules that physicists only began to understand in the last century, yet evolution discovered these principles billions of years before. The tunneling times, measured in picoseconds, are shorter than the duration of almost any thought or perception, and yet within these unimaginably brief intervals, the precise positioning of atoms and the careful tuning of energy levels that evolution has achieved make all the difference between efficient energy capture and wasteful energy loss. There is no drama in these quantum jumps, no struggle or striving, merely the smooth unfolding of physical law in molecular structures refined over geological time. And with that gentle thought, of electrons taking their quantum shortcuts through barriers that classical physics would call impassable, we can let our own thoughts grow soft and unfocused, trusting that the physics of the quantum world will continue its work whether we are awake to contemplate it or drifting peacefully toward sleep. Let us turn our attention now to the deep history of photosynthesis, a story that stretches back nearly 3 billion years into the ancient past of our planet, and as you settle into the comfortable rhythm of these words, you may find it rather soothing to consider how the very oxygen that fills your lungs with each slow breath is the product of an unbroken chain of quantum processes that began long before any complex life existed on Earth, when our world was a strange and inhospitable place, shrouded in an atmosphere that would be quite toxic to us today, yet somehow perfectly suited to the microscopic pioneers that would eventually transform everything. The Earth that existed before widespread photosynthesis was remarkably different from the blue and green world we know, with an atmosphere composed primarily of nitrogen, carbon dioxide, methane, and water vapor, containing almost no free oxygen whatsoever, and the skies would have appeared hazy, perhaps tinged with orange or pink from the methane that drifted through the upper atmosphere, while the oceans themselves may have carried a greenish tint from dissolved iron compounds that had not yet been oxidized, and it is quite peaceful to imagine this quiet, alien world slowly turning beneath its young sun, waiting without any awareness for the chemical revolution that was already beginning in its warm, shallow seas. The Great Oxygenation Event, which occurred approximately 2.4 billion years ago, represents one of the most significant transformations in the history of our planet, a gradual but profound shift in the composition of Earth's atmosphere that was driven entirely by the patient, ceaseless work of photosynthetic microorganisms, and this event, sometimes called the oxygen catastrophe because it was indeed catastrophic for the anaerobic life forms that had dominated until then, marked the moment when oxygen produced by photosynthesis finally overwhelmed the various chemical sinks that had been absorbing it, allowing free oxygen to accumulate in the atmosphere for the first time. And you might find it rather calming to reflect on how this transformation happened not through any dramatic upheaval but through the quiet, repetitive splitting of water molecules, one photon at a time, across countless generations of single-celled organisms. The evidence for these ancient photosynthetic communities comes to us from the patient work of geologists and paleontologists, who have learned to read the subtle signatures preserved in very old rocks, and one particularly important discovery was made by Elso Barghorn, a paleobotanist working at Harvard University, who in 1954 began his detailed examination of the Gunflint Chert, a formation of silica-rich rock found along the northern shores of Lake Superior in Ontario, Canada, and within these ancient stones, which had formed approximately 1.9 billion years ago when silica-laden water had percolated through microbial mats and preserved them with exquisite fidelity, Barghorn and his colleague Stanley Tyler discovered beautifully preserved microfossils that bore a striking resemblance to modern cyanobacteria, those remarkable organisms that first mastered the art of oxygenic photosynthesis. The gunflint fossils, which Barghorn described in a series of careful publications throughout the 1950s and 60s, included filamentous structures and spherical cells that showed clear evidence of biological origin, with some specimens preserving details as fine as the internal organization of the ancient cells, and these discoveries helped establish that complex microbial communities, including photosynthetic organisms capable of the quantum processes we have been discussing, were flourishing on Earth nearly 2 billion years ago, and the gentle preservation of these tiny beings in their silica tombs offers us a window into a world where the fundamental chemistry of photosynthesis was already well established, the light harvesting machinery already refined through hundreds of millions of years of evolution. But where did the chloroplasts in modern plant cells come from? Those tiny green organelles that contain all the molecular machinery for capturing light and converting it into chemical energy. And the answer to this question leads us to one of the most elegant theories in biology, proposed by Lynn Margulis in a paper published in 1967 in the Journal of Theoretical Biology, a paper that was rejected by approximately 15 journals before finally finding a home. So controversial was its central premise at the time, and Margulis, working then under her married name Lynn Sagan, suggested that chloroplasts, along with mitochondria, were not structures that had evolved gradually within cells but were instead the descendants of free-living bacteria that had been engulfed by larger cells in the distant past, entering into a symbiotic relationship so intimate that they eventually became permanent residents, unable to survive independently. This endosymbiotic theory, as it came to be known, proposed that the ancestors of modern plant cells had essentially swallowed cyanobacteria, those same photosynthetic microorganisms whose fossils Barghorn had found in the Gunflint Chert, And rather than digesting these captive bacteria, the host cells had allowed them to persist, benefiting from the sugars produced through photosynthesis while providing the cyanobacteria with protection and access to nutrients. And over vast stretches of evolutionary time, measured in hundreds of millions of years, the captured bacteria lost many of their genes, transferring them to the nucleus of the host cell, while retaining just enough genetic material and cellular machinery to carry out their essential function of harvesting light. And this theory, initially met with considerable skepticism, has since been supported by overwhelming evidence from multiple lines of inquiry. The genetic evidence for endosymbiosis is particularly compelling, and you may find it rather soothing to consider how the quiet work of molecular biologists in the decades following Margulis's original proposal gradually revealed the truth of her hypothesis, because when scientists sequenced the DNA found within chloroplasts, they discovered that it bears a remarkable resemblance to the genomes of modern cyanobacteria, with similar gene arrangements, similar regulatory sequences, and even similar mechanisms for replicating and expressing genetic information. And the chloroplast genome is organized in a circular chromosome, just as bacterial genomes are, rather than the linear chromosomes found in the nuclei of eukaryotic cells, and the ribosomes within chloroplasts, those tiny molecular machines that translate genetic information into proteins, are more similar to bacterial ribosomes than to the ribosomes found elsewhere in plant cells, responding to the same antibiotics that affect bacteria while remaining unaffected by drugs that target eukaryotic ribosomes. The evolution of photosynthesis itself, from its earliest origins to the sophisticated quantum machinery found in modern plants, represents a journey of remarkable refinement. And the best current estimates suggest that oxygenic photosynthesis, the type that splits water and releases oxygen, first evolved approximately 2.7 billion years ago, some 300 million years before the Great Oxygenation Event. And this gap between the origin of oxygen-producing photosynthesis and the actual oxygenation of the atmosphere can be explained by the presence of various chemical sinks that absorbed the oxygen as quickly as it was produced, including dissolved iron in the oceans that reacted with oxygen to form the banded iron formations that geologists find in ancient rocks around the world. And only when these sinks were finally saturated did oxygen begin to accumulate in the atmosphere, a process that unfolded over millions of years with the gentle inevitability of water slowly filling a basin. Before oxygenic photosynthesis, there existed simpler forms of the process, known as anoxygenic photosynthesis, which used molecules other than water as the source of electrons, including hydrogen sulfide, hydrogen gas, or various organic compounds, and these earlier forms of photosynthesis did not produce oxygen as a byproduct, and the organisms that carried out these reactions, ancestors of modern purple bacteria and green sulfur bacteria, possessed photosynthetic machinery that was simpler than the two-photosystem arrangement found in cyanobacteria and plants. And understanding how the more complex oxygenic system evolved from these simpler precursors has been a major focus of research in evolutionary biology. In 2004, John Raymond and Robert Blankenship published a phylogenetic analysis in the journal Coordination Chemistry Reviews that shed considerable light on this question, examining the evolutionary relationships between different types of photosynthetic reaction centers and tracing their origins through careful comparison of genetic sequences, and their work revealed something quite remarkable, that the two photosystems found in oxygenic photosynthesis, known as photosystem I and photosystem II, appear to have separate evolutionary origins, deriving from different lineages of anoxygenic photosynthetic bacteria, and the combination of these two systems into a single organism, creating the Z-scheme of electron transport that allows water to be split and oxygen to be released, appears to have involved horizontal gene transfer, a process by which genetic material is exchanged between organisms that are not parent and offspring, allowing evolutionary innovations to spread across different lineages of bacteria. This finding has profound implications for our understanding of how the quantum machinery of photosynthesis came to be, suggesting that evolution acted as a kind of tinkerer, combining components from different sources to create something more powerful than either could achieve alone, and the horizontal gene transfer that brought these systems together would have occurred in an ancient world of microbial mats and shallow seas, where different types of bacteria lived in close proximity, exchanging genetic material through processes that continue in bacterial communities today, and the result of this genetic mixing was the emergence of a photosynthetic system capable of using the most abundant molecule on Earth, water, as its source of electrons, opening up ecological possibilities that had previously been inaccessible. The question of whether evolution specifically selected for quantum effects in photosynthesis leads us to a fascinating concept known as quantum Darwinism, though we should note that this term is used in slightly different ways by different researchers, and in one sense it refers to the broader question of whether natural selection can act on quantum mechanical properties of biological systems, favoring organisms whose molecular machinery makes better use of quantum phenomena such as coherence and tunneling, and the remarkable efficiency of photosynthetic energy transfer, which appears to exploit quantum effects to achieve near-perfect performance, suggests that evolution may indeed have optimized these quantum properties over billions of years, selecting for molecular arrangements that maintain quantum coherence just long enough to enhance energy transfer without requiring the extreme conditions of temperature and isolation that physicists typically use to study quantum systems in the laboratory. Living fossils of these ancient photosynthetic communities can still be found in certain protected environments around the world, and perhaps the most famous of these are the stromatolites of Shark Bay in Western Australia, where colonies of cyanobacteria build layered structures of sediment and organic material that closely resemble the fossilized stromatolites found in rocks billions of years old, and these living stromatolites grow in the hypersaline waters of Hamelin Pool, where the high salt concentration excludes the grazing animals that would otherwise consume the microbial mats, allowing the cyanobacteria to build their characteristic dome-shaped structures just as their ancestors did in the Precambrian seas, and visiting scientists describe these formations with a sense of quiet wonder, standing in the presence of organisms whose basic biochemistry has remained fundamentally unchanged across an almost incomprehensible span of time. One of the most remarkable aspects of photosynthesis, as revealed by studies comparing different organisms across the tree of life, is how consistently efficient the process has remained throughout its evolutionary history, with the fundamental quantum yield of photosynthesis, the number of oxygen molecules produced per photon absorbed, remaining close to the theoretical maximum across a wide range of organisms, from ancient cyanobacteria to modern flowering plants, and this consistency suggests that the basic machinery of photosynthesis was optimized very early in its evolutionary history and has been maintained by natural selection ever since, with variations occurring primarily in the light-harvesting antenna systems that collect photons and funnel their energy to the reaction centers, while the reaction centers themselves have remained remarkably conserved. The quantum processes at the heart of photosynthesis, the delicate superpositions and the coherent energy transfer and the precise molecular arrangements that make it all possible, have thus been operating continuously for approximately 3 billion years, through ice ages and warm periods, through mass extinctions and evolutionary radiations, Through the colonization of land by plants and the subsequent development of forests and grasslands, and every green leaf you have ever seen, every blade of grass, every floating speck of algae in pond or ocean, contains molecular machinery whose basic design was established before any animal had evolved, before any multicellular organism existed, in a world of single cells and shallow seas and hazy orange skies slowly turning blue. As you rest now and allow these thoughts to settle gently in your mind, you might reflect on the extraordinary continuity that connects you to those ancient cyanobacteria, not merely through the oxygen they produced but through the very chloroplasts in every plant cell, descendants of bacteria that were captured and domesticated so long ago, and the quantum dance of energy transfer that occurs in every leaf outside your window, should there be one, follows choreography that was refined over billions of years of patient evolutionary tinkering, selecting for arrangements of molecules that could harvest light with exquisite efficiency, maintaining quantum coherence in the warm, wet environment of a living cell, a feat that still challenges our best attempts at artificial replication, and this ancient machinery continues its quiet work as you settle deeper into rest, splitting water, releasing oxygen, capturing the energy of sunlight, one photon at a time, as it has done since long before there were eyes to see the green of leaves or minds to wonder at the quantum strangeness that makes it all possible. And so we come now to a rather different chapter in this story, one that moves us gently from the quiet interior of leaves and the strange behaviors of molecules into the busy world of laboratories and research institutions, where scientists have spent decades trying to understand and recreate what plants accomplish so effortlessly every moment of every day. This is the story of artificial photosynthesis, the patient and methodical effort to build human-made systems that can do what chlorophyll and proteins and carefully arranged molecular structures have been doing for billions of years, which is to say, capturing sunlight and storing its energy in chemical bonds that can be used later, whenever and wherever that energy might be needed. The story of this scientific pursuit is, in many ways, a story of humility, because the more researchers learned about how photosynthesis actually works at the quantum mechanical level, the more they came to appreciate just how remarkably sophisticated nature's solution really is, and how challenging it would be to replicate even a small portion of that elegance in artificial systems. But it is also a story of persistence and gradual progress, of small victories accumulating over years and decades, and of the slow refinement of materials and designs that bring us incrementally closer to machines that might one day harvest sunlight as effectively as the leaves outside your window. One significant milestone in this long journey came in the year 2010, when the United States Department of Energy established the Joint Center for Artificial Photosynthesis, an institution created specifically to coordinate and accelerate research into these technologies. The center received initial funding of $122 million, a substantial investment that reflected the growing recognition that artificial photosynthesis might one day play an important role in addressing energy challenges. The Joint Center was established as a partnership between two of America's most distinguished research institutions, the California Institute of Technology in Pasadena and the Lawrence Berkeley National Laboratory across the bay from San Francisco, bringing together chemists and physicists and materials scientists and engineers under one organizational umbrella, all working toward the common goal of building systems that could convert sunlight into fuel. The center represented something of a new approach to this kind of research, an acknowledgement that the problem was too large and too complex for any single laboratory to solve in isolation, and that progress would require the kind of sustained, coordinated effort that only major institutional support could provide. Researchers at both locations began working on different aspects of the challenge, some focusing on the light-absorbing components, others on the catalysts that would drive the chemical reactions, still others on the membranes and interfaces that would need to separate products and manage the flow of electrons and protons through the system. Around this same time, in 2011, a chemist named Daniel Norsera, who was then working at the Massachusetts Institute of Technology, demonstrated something he called an artificial leaf, a device that captured considerable public attention because of its elegant simplicity and its evocative name. Norsera's artificial leaf was a thin wafer of silicon, not much larger than a playing card, coated with special catalysts that could split water molecules into hydrogen and oxygen when exposed to sunlight. You could drop this wafer into a glass of water, shine light upon it, and watch as bubbles of gas began to form on its surface, hydrogen on one side and oxygen on the other, the same basic reaction that occurs in the oxygen-evolving complex of photosystem II, though achieved through rather different chemical means. What made Norsera's work particularly noteworthy was his choice of catalyst. For the oxygen-evolving side of the reaction, he used a material based on cobalt and phosphate, elements that are relatively abundant and inexpensive compared to the precious metals that many earlier artificial photosynthesis systems had required. The catalyst could form spontaneously from a solution containing cobalt ions and phosphate, assembling itself on the electrode surface under the right conditions, and it could operate in ordinary water at neutral pH, which is to say, water that is neither particularly acidic nor particularly basic, the kind of water you might find in a pond or a stream or indeed in the glass beside your bed. This question of materials has been, and continues to be, one of the central challenges in the field of artificial photosynthesis. Nature, you see, builds her photosynthetic machinery from elements that are abundant and easily obtained, the kind of atoms that were readily available in the ancient oceans where photosynthesis first evolved. The oxygen-evolving complex at the heart of photosystem II, that remarkable cluster we discussed earlier, is built from manganese and calcium and oxygen, elements that are common in the Earth's crust and dissolved in seawater, elements that biology could access without difficulty. The light-harvesting antennae are built from magnesium and nitrogen and carbon, woven into the porphyrin rings of chlorophyll molecules. Even the iron-sulfur clusters that transfer electrons through the photosynthetic chain are made from two of the most abundant elements on our planet. Early artificial photosynthesis systems, however, often required platinum and ruthenium and iridium, elements that are exceedingly rare in the Earth's crust, elements that are expensive precisely because they are so scarce. Platinum, for instance, is an excellent catalyst for many reactions, including the production of hydrogen gas, but its rarity means that any technology requiring large amounts of platinum would be difficult to scale up to the levels needed to make a meaningful contribution to global energy supplies. The challenge, then, has been to find catalysts made from Earth-abundant materials that can perform as well as, or nearly as well as, these precious metal catalysts, and this is where Norsera's cobalt phosphate work represented such an encouraging step forward. The effort to mimic photosynthesis artificially has roots that stretch back well before the establishment of the Joint Center or Norsera's artificial leaf demonstration. At Arizona State University, researchers Devons Gust and Thomas Moore, along with their colleague Anna Moore, have been working on artificial photosynthesis systems since the 1980s, building their designs around porphyrin molecules, those same ring-shaped structures that form the core of chlorophyll. Porphyrins have certain properties that make them particularly attractive for light harvesting, including their ability to absorb light strongly and their relative stability under illumination. And Gust and Moore and their collaborators spent years learning how to link porphyrins together with other molecular components to create systems that could absorb light and transfer energy and separate charges, mimicking in simplified form the processes that occur in natural photosynthetic reaction centers. Their work helped establish what researchers sometimes call the antenna reaction center paradigm, a design philosophy for artificial photosynthesis that takes direct inspiration from how plants and photosynthetic bacteria organize their molecular machinery. In natural systems, as we have discussed, large arrays of light-harvesting pigments, the antenna, absorb photons and funnel the resulting excitation energy toward a reaction center, where the actual charge separation occurs. This arrangement allows the organism to capture light over a broad area while concentrating the harvested energy at the specific location where the chemistry happens. The artificial systems designed by Gust and Moore and others attempt to recreate this same basic architecture, with synthetic antenna molecules absorbing light and passing energy to synthetic reaction center components where electrons can be moved from one place to another. The efficiency with which natural and artificial systems perform these tasks is a subject worth considering for a moment, because the numbers reveal something interesting about the different ways we might measure success. When we speak of efficiency in photosynthesis, we need to be careful about what exactly we are measuring. The overall efficiency with which plants convert sunlight into biomass, into the actual stuff of leaves and stems and roots, is really quite low, typically around 1% or even less for most plants under normal growing conditions. This might seem surprisingly modest for a process that has had billions of years to evolve, but it reflects the many steps between initial light absorption and final incorporation of carbon into plant tissue, each step with its own inefficiencies. And it reflects the fact that plants have been optimized by evolution not just for energy capture but for survival and reproduction under variable and often challenging conditions. But if we look instead at just the initial light harvesting step, at the efficiency with which absorbed photons are converted into separated charges within the reaction center, the numbers are remarkably different. This initial quantum efficiency can be as high as 95% or even higher, meaning that nearly every photon absorbed by the antenna system successfully produces a charge-separated state in the reaction center. This is the aspect of photosynthesis that has most captivated physicists and chemists, this near-perfect efficiency in the earliest stages of energy conversion. And it is this efficiency that artificial systems attempt to approach. Current artificial photosynthesis systems have achieved solar-to-fuel efficiencies of around 10% in some cases, which is considerably higher than the overall efficiency of natural biomass production but still leaves room for improvement, particularly when you consider that some of the theoretical limits for such systems are considerably higher. The goal is not necessarily to beat nature at her own game but rather to build systems that can operate with high enough efficiency and low enough cost to compete with other ways of producing fuel. Nathan Lewis, a chemist at the California Institute of Technology who has been deeply involved with the Joint Center for Artificial Photosynthesis, has pursued approaches that combine semiconductors with liquid solutions, creating what are called semiconductor-liquid junctions where light absorption in the solid material can drive chemical reactions in the adjacent liquid. This approach draws on technologies developed for solar cells, which have become increasingly efficient and inexpensive over the past several decades, and attempts to extend them to the production of fuels rather than electricity. Lewis and his collaborators have worked on developing materials that can absorb sunlight efficiently, that can catalyze the necessary chemical reactions at their surfaces, and that can remain stable when immersed in water or other liquids for extended periods, which has proven to be one of the more difficult technical challenges. The practical goal that guides much of this research can be stated in fairly concrete terms: to produce hydrogen fuel at a cost of $2 to $4 per kilogram, which is the price range at which hydrogen might become competitive with fossil fuels for various applications. Hydrogen is an attractive fuel in many ways, because when it burns or reacts in a fuel cell, the only product is water, with none of the carbon dioxide emissions that accompany the burning of coal or oil or natural gas. But hydrogen does not exist freely in nature in significant quantities. It must be produced from other substances, currently most often from natural gas, through a process that releases carbon dioxide, which rather defeats the environmental purpose. Producing hydrogen from water using only sunlight as an energy input would be a genuinely clean source of fuel, but only if the systems that accomplish this feat can be built cheaply enough and operate efficiently enough to make the resulting hydrogen economically viable. The $2 to $4 per kilogram target represents a significant challenge. Current methods of producing hydrogen from water using electricity, a process called electrolysis, typically produce hydrogen at higher costs, and the electricity itself must come from somewhere. An artificial photosynthesis system that could take in only sunlight and water and produce hydrogen at this target cost would represent a considerable technological achievement, and researchers around the world continue to work toward this goal, making incremental progress in catalyst development, in materials science, in system integration, and in our fundamental understanding of how to make artificial systems that capture and convert light as elegantly as the leaves that have surrounded us since long before we were here to notice them. The research continues in laboratories in California and Arizona and Massachusetts and many other places around the world, patient work that proceeds one experiment at a time, one publication at a time, one small improvement at a time. The scientists involved understand that they are working on a long-term problem, one that may take decades more to solve completely, but they are sustained by the knowledge that nature has already demonstrated that the task is possible, that it can be done with abundant materials and at ambient temperatures, and that the secrets of how it is accomplished are there to be discovered by those with the patience and skill to look closely enough at what happens when sunlight falls upon a leaf. And so we arrive at a chapter in our story that concerns not the molecules themselves, not the delicate dance of energy through protein and pigment, but rather the human beings who have devoted their careers to understanding these processes, and the very human disagreements that have shaped our understanding of quantum biology. For in science, as in so many fields of human endeavor, the path to truth is rarely straight, and reasonable people examining the same evidence can arrive at quite different conclusions, and this is not a failing of the scientific method but rather one of its essential features, the way in which competing ideas are tested and refined through careful argument and ever more precise experiment. The story of quantum effects in photosynthesis is also a story of scientific controversy, of skeptics and believers, of heated debates at conferences and pointed exchanges in journal pages, all conducted with the underlying shared commitment to understanding how nature actually works, even when that understanding proves elusive and contentious. The excitement that followed the 2007 Berkeley experiments, those landmark observations of quantum coherence in bacterial light harvesting systems, naturally gave rise to questions and doubts among those scientists who had spent decades studying photosynthesis through more traditional frameworks. Among the most influential skeptical voices were those of Paul Broomer and Moshe Shapiro, distinguished theoretical chemists whose 2012 paper cast significant doubt on the claim that quantum coherence actually aids the efficiency of energy transfer. Their argument, carefully constructed and mathematically rigorous, pointed out that the conditions under which coherence had been observed in laboratory experiments bore little resemblance to the conditions experienced by actual living organisms. The ultra-short laser pulses used to excite the photosynthetic complexes created a very specific type of quantum superposition, a coherent excitation in which multiple pigment molecules were excited simultaneously and in phase with one another. But sunlight, Broomer and Shapiro noted, is nothing like an ultra-short laser pulse. Natural sunlight is incoherent, arriving at the photosynthetic apparatus in a continuous, random fashion, with photons arriving independently rather than in the precisely timed bursts produced by laboratory lasers. This distinction matters enormously, the skeptics argued, because the coherence observed in the Berkeley experiments and their successors might simply be an artifact of the excitation conditions, a reflection of the coherent laser light rather than an intrinsic property of the biological system under natural conditions. If you excite a system coherently, they pointed out, you should not be surprised to observe coherent dynamics in the immediate aftermath, but this tells you relatively little about how the system behaves when excited by the incoherent light that plants and bacteria actually encounter in nature. The Broomer-Shapiro critique cut to the heart of the quantum biology excitement, suggesting that while the experiments were undoubtedly technically impressive and scientifically interesting, they might not reveal anything particularly relevant about how photosynthesis actually works in living organisms going about their daily business of converting sunlight into chemical energy. The year before this influential paper appeared, in March of 2011, these tensions came to a head at the annual meeting of the Biophysics Society, held that year in Baltimore, Maryland. The conference brought together experimentalists and theorists from around the world, and sessions devoted to quantum effects in biology became focal points for intense discussion. Researchers who had observed long-lived coherence presented their latest findings, while skeptics challenged the interpretation of those findings, questioning whether the observed effects were genuinely functional or merely incidental byproducts of molecular dynamics with no particular biological significance. The debates were vigorous, as scientific debates should be, with neither side quite convincing the other but with both sides clarifying their positions and identifying the key questions that would need to be answered by future research. Some participants left Baltimore more convinced than ever that quantum mechanics played a crucial role in photosynthesis, while others left equally convinced that the quantum coherence community had overinterpreted their data and drawn conclusions that went far beyond what the evidence could support. In the years that followed, various researchers attempted to develop theoretical frameworks that might reconcile these competing viewpoints, and among the most intriguing of these was the concept of vibration-assisted coherence, proposed and developed by Alexandra Olaya-Castro and her collaborators. Olaya-Castro, a theoretical physicist whose work bridges quantum physics and biology, suggested that the protein environment surrounding the photosynthetic pigments might not simply be a passive scaffold but an active participant in the energy transfer process. Her theory proposed that specific vibrational modes of the protein and pigment molecules could become entangled with the electronic excitations responsible for energy transfer, creating a form of coherence that was neither purely electronic nor purely vibrational but a hybrid of the two. This vibration-assisted coherence, the theory suggested, could survive much longer than purely electronic coherence and could actually be generated and sustained even under incoherent excitation conditions, potentially addressing some of the criticisms raised by Broomer and Shapiro. The vibration-assisted theory made specific predictions that could be tested experimentally, including the expectation that certain vibrational frequencies of the pigment-protein complex would show correlations with the electronic energy levels that could not be explained by classical mechanics alone. If specific vibrations were indeed coupled to the electronic excitation in the manner the theory described, then modifying those vibrations, either through isotope substitution or through chemical modification of the protein, should produce measurable changes in the energy transfer dynamics. These predictions provided a roadmap for future experiments, a way to distinguish between competing theoretical frameworks by designing measurements whose outcomes would differ depending on which framework was correct. Meanwhile, at the Max Planck Institute for the Structure and Dynamics of Matter in Hamburg, Duane Miller and his colleagues were developing new experimental techniques that promised to provide unprecedented insight into the ultrafast dynamics of molecular systems, including photosynthetic complexes. Miller's group specialized in ultrafast electron diffraction, a technique that uses extremely short pulses of electrons, lasting only femtoseconds, to take snapshots of molecular structure during chemical reactions and physical processes. Unlike optical spectroscopy, which provides information primarily about electronic states, electron diffraction reveals the positions of atoms directly, allowing researchers to watch the actual structural changes that accompany energy transfer and other molecular dynamics. By applying these techniques to photosynthetic systems, Miller hoped to observe not just the electronic coherence that had been the focus of so much attention but also the nuclear motions, the vibrations and conformational changes of the protein and pigment molecules that might be coupled to the electronic dynamics in the manner suggested by the vibration-assisted coherence theory. As experimental techniques improved and theoretical understanding deepened, a more nuanced picture began to emerge, one in which the early enthusiasm for quantum coherence was tempered by careful consideration of artifacts and alternative explanations. Critics pointed out that the characteristics of the ultra-short laser pulses used in the early experiments could themselves influence the observed dynamics in subtle ways that might be mistaken for intrinsic properties of the biological system. The spectral width of the laser pulses, their temporal shape, their intensity, all of these parameters could potentially create artifacts that would appear as coherent oscillations in the experimental signal even if no such oscillations existed in the actual molecular system. Careful control experiments and improved data analysis techniques were needed to distinguish genuine biological coherence from experimental artifacts, and as researchers became more sophisticated in their approach, some of the early claims were revised or qualified. Theoretical work by Martin Plinio and his group at the University of Ulm provided another important piece of the puzzle through their development of the concept of environment-assisted quantum transport. Conventional wisdom in quantum physics had long held that interaction with the environment, the phenomenon of decoherence, was purely destructive, serving only to destroy delicate quantum superpositions and return systems to classical behavior. But Plinio's work showed that under certain circumstances, just the right amount of environmental noise could actually enhance transport efficiency rather than diminishing it. The environment, in this view, serves not as an enemy of quantum coherence but as a kind of lubricant that helps energy find its way through complex molecular networks. Too little environmental interaction leaves the excitation trapped in localized states, unable to explore the full energy landscape, while too much environmental interaction causes the quantum coherence to collapse before it can be useful. But at intermediate levels of environmental coupling, the noise assists rather than hinders transport, a phenomenon that came to be known by various names including noise-assisted transport and environment-assisted quantum transport. This theoretical framework suggested that photosynthetic organisms might have evolved to operate in precisely this intermediate regime, where quantum coherence and environmental noise work together to achieve efficient energy transfer. The protein environment, far from being an obstacle to quantum effects, might be specifically tuned to support them, providing just enough fluctuation to assist transport while not so much as to destroy the coherence entirely. This perspective helped to explain how quantum effects could survive in the warm, wet, noisy environment of living cells, a question that had puzzled researchers since the first observations of long-lived coherence in photosynthetic systems. By 2020, nearly 15 years after the first compelling observations of quantum coherence in photosynthesis, the field had matured sufficiently that a comprehensive assessment of the evidence seemed both possible and necessary. In that year, a major review article appeared in Science Advances, authored by Jianshu Cao, Robert Silby, and numerous colleagues who had been central figures in the quantum biology debates. This review attempted to synthesize the accumulated evidence from more than a decade of experiments and theoretical work, providing a balanced assessment of what had been learned and what remained uncertain. The authors acknowledged that quantum coherence had been convincingly demonstrated in photosynthetic systems under laboratory conditions, but they were more cautious about claims that such coherence was functionally important for the efficiency of photosynthesis in living organisms. The review highlighted the need for experiments under more physiologically relevant conditions and for theoretical models that could make quantitative predictions about the functional consequences of quantum effects. Later that same year, in November of 2020, a paper appeared in Nature Chemistry that attempted to reframe the entire debate in a way that might allow the field to move forward constructively. Authored by Cao and several colleagues, the paper bore the provocative title of Quantum Biology Without Quantum Weirdness, and it argued that much of the controversy surrounding quantum effects in biology had been fueled by unnecessary mystification and imprecise language. The authors suggested that the phenomena observed in photosynthesis, while undoubtedly quantum mechanical in origin, did not require invoking the stranger aspects of quantum theory that captured the popular imagination, phenomena like quantum entanglement or wave function collapse or the observer-dependent nature of quantum measurement. Instead, they proposed that the relevant quantum effects could be understood in terms of more mundane quantum phenomena, the wave-like properties of matter, the interference effects that arise from coherent superpositions, the discrete energy levels of molecular systems. These effects, while quantum mechanical, were not particularly mysterious or counterintuitive, at least not to physicists who had spent their careers working with quantum systems in other contexts. The quantum biology without quantum weirdness perspective represented a kind of middle ground between the most enthusiastic proponents of quantum biology and the most skeptical critics. It accepted that quantum mechanics was essential for understanding energy transfer in photosynthesis, in the same way that quantum mechanics is essential for understanding the behavior of any molecular system, but it rejected the more extravagant claims that photosynthesis somehow harnessed exotic quantum phenomena in ways that transcended ordinary quantum chemistry. This reframing allowed researchers to focus on the substantive scientific questions: how energy flows through photosynthetic complexes, what role coherent dynamics play in that flow, how the molecular structure of these systems has been shaped by evolution, without getting distracted by debates about whether the phenomena under study qualified as sufficiently weird to merit special attention. And so the field continues, neither fully resolved nor abandoned, as scientific fields so often do, with new experiments and new theories gradually refining our understanding of these beautiful molecular machines that have been quietly harvesting sunlight for billions of years. The controversies, heated though they sometimes were, served the essential purpose of forcing researchers to think more carefully about their assumptions, to design more rigorous experiments, to develop more sophisticated theoretical frameworks. Whether quantum coherence proves to be functionally important for photosynthesis or merely an incidental byproduct of molecular physics, the effort to understand these systems has deepened our knowledge of both quantum mechanics and biology, and that knowledge, accumulated through the patient work of many researchers over many years, represents a kind of truth that transcends any particular debate or controversy, a truth that we continue to approach, step by careful step, as we drift now toward sleep and rest. And so we arrive now at a rather lovely turning point in our story, where the quiet discoveries made in plant biology laboratories begin to ripple outward into the world of computing, where the lessons learned from chlorophyll and protein scaffolds find their way into the design of machines that may one day solve problems beyond our current imagining, and where nature's ancient solutions to energy harvesting become templates for humanity's newest technologies, all of which unfolds with the same unhurried patience that characterizes the growth of a leaf toward sunlight. The connection between photosynthesis and quantum computing might seem, at first consideration, rather unexpected, for one involves living organisms that have been quietly going about their business for billions of years, while the other represents cutting-edge human engineering with supercooled circuits and precisely controlled electromagnetic fields, and yet upon closer examination, the relationship reveals itself to be both natural and deeply instructive, as researchers have come to recognize that plants and certain bacteria solved problems of quantum efficiency long before we even knew such problems existed, and that their solutions, refined through countless generations of evolutionary trial, offer insights that no amount of theoretical work alone might have provided. The story of this intellectual cross-pollination begins in earnest with the work of Seth Lloyd, a physicist at the Massachusetts Institute of Technology who has spent much of his career thinking about the fundamental nature of computation and the quantum mechanical processes that underlie all physical reality, and in a paper published in 2011, Lloyd and his collaborators proposed something rather remarkable, namely that photosynthesis performs what might reasonably be called a natural quantum computation, using the principles of quantum mechanics not merely incidentally but in a manner that achieves a functional outcome, the efficient transfer of energy, through processes that bear striking resemblance to algorithms designed by human quantum computer scientists. Lloyd's insight rested upon recognizing that the energy transfer problem faced by photosynthetic systems bears structural similarity to certain computational search problems, particularly the challenge of finding a specific item within a large unsorted database, which is precisely the problem addressed by Grover's search algorithm, one of the most celebrated achievements of quantum computing theory, developed by Love Grover in 1996, which demonstrated that quantum computers could search through unsorted databases quadratically faster than any classical computer, and Lloyd observed that nature appeared to have discovered something analogous, using quantum coherence to sample multiple pathways simultaneously in a manner that finds the optimal energy transfer route with remarkable efficiency. The mathematical framework connecting these two domains involves what are known as quantum walks, which represent the quantum mechanical generalization of classical random walks, those processes by which a particle or entity moves through a space by taking steps in random directions, and while a classical random walk explores its environment in a diffusive, gradually spreading manner, A quantum walk exhibits interference effects that can dramatically accelerate the exploration of certain structures, spreading quadratically faster through regular lattices and exploring graph structures with an efficiency that depends intimately on the geometry of the space being traversed, and it is precisely this quantum walk behavior that researchers have identified in the energy transfer dynamics of photosynthetic complexes, where excitons appear to perform something very much like a quantum walk across the network of chromophores embedded within their protein matrices. Now, one might reasonably ask how such a connection could be practically useful, for understanding that nature performs quantum computations is intellectually satisfying but does not automatically translate into better quantum computers. And here we encounter one of the most surprising and counterintuitive discoveries to emerge from photosynthesis research, a finding that challenged assumptions held by quantum computer scientists and suggested new approaches to quantum algorithm design, namely the phenomenon known as noise-assisted transport, or in its more formal theoretical framework, environment-assisted quantum transport, abbreviated as ENACT. The discovery of noise-assisted transport emerged from the theoretical work of Martin Plinio and Susanna Huelger, physicists working at Ulm University in Germany who had been studying the quantum dynamics of photosynthetic systems, and what they found was rather contrary to expectations, for conventional wisdom in quantum computing held that environmental noise was purely detrimental to quantum processes, that decoherence represented an enemy to be fought with ever more sophisticated error correction and isolation techniques, and yet Plinio and Huelger demonstrated through careful theoretical analysis that in photosynthetic systems, a certain amount of environmental noise actually enhanced energy transfer efficiency, helping to overcome what might otherwise be destructive interference patterns and assisting the exciton in reaching the reaction center more reliably than would occur in a perfectly isolated quantum system. The ENACT framework provides a mathematical description of this phenomenon, explaining how the interplay between quantum coherent dynamics and environmental decoherence can be optimized to achieve transfer efficiencies exceeding what either purely quantum or purely classical mechanisms could achieve alone, and the key insight is that too much coherence can be just as problematic as too little, for in a perfectly coherent system, destructive interference can trap energy in localized states, preventing it from reaching its intended destination, while just the right amount of environmental noise serves to disrupt these interference patterns, allowing the energy to escape such traps and continue its journey toward the reaction center. And nature, through billions of years of evolution, has tuned this balance with exquisite precision. This discovery has profound implications for quantum computing, suggesting that the traditional approach of maximizing isolation and minimizing decoherence may not always be optimal, and that certain computational tasks might actually benefit from a controlled amount of noise, a possibility that has opened new avenues for quantum algorithm design and quantum hardware engineering, where researchers now consider not only how to eliminate environmental interference but also how to harness it constructively, learning from the photosynthetic organisms that mastered this balance long ago. The collaboration between quantum computing researchers and photosynthesis scientists has taken concrete institutional form, with teams at major technology companies and research laboratories working together to translate biological insights into computational advances, and IBM's quantum computing division has been particularly active in this regard, bringing together experts in quantum biology and quantum information science to explore the connections between natural and artificial quantum systems, seeking to understand whether the architectural principles that make photosynthesis so efficient might inform the design of quantum processors and the algorithms that run upon them. At Google, this interdisciplinary work has been championed by researchers including Masoud Mohini, a physicist whose career has spanned both quantum biology and quantum machine learning, and who has been instrumental in developing frameworks for understanding how biological quantum systems process information, with particular attention to how these insights might enhance artificial intelligence and machine learning algorithms. Recognizing that nature's solution to energy harvesting may contain lessons applicable to a surprisingly wide range of computational challenges, from optimization problems to pattern recognition to the training of neural networks. Google's quantum supremacy demonstration in 2019 provides a useful reference point for understanding the potential significance of these biological insights, for in that experiment, a quantum processor called Sycamore performed a specific computational task, involving the sampling of random quantum circuits, in approximately 200 seconds, while the researchers estimated that the most powerful classical supercomputers would require approximately 10,000 years to perform the equivalent calculation, and while this particular task has no immediate practical application, the demonstration established that quantum computers can indeed outperform classical machines for certain problems, opening the door to the possibility that quantum advantages might be achieved for more practically relevant computations. The conceptual connection to photosynthesis lies in the nature of the computational advantage, for both the quantum supremacy experiment and photosynthetic energy transfer derive their efficiency from the same fundamental quantum mechanical principles, the superposition of states and the interference of probability amplitudes, which allow exploration of exponentially large spaces in ways that classical systems cannot match, and while the specific problems are quite different, harvesting light energy versus sampling random circuits, the underlying physics shares important features, suggesting that insights gained in one domain may well illuminate the other. The United States Department of Energy recognized the strategic importance of these connections in 2018, when it established quantum computing initiatives that explicitly cited photosynthesis research as a source of inspiration and insight, acknowledging that the billions of years of evolutionary refinement embodied in biological quantum systems represent an invaluable resource for guiding human engineering efforts and funding research programs that bring together biologists, physicists, and computer scientists to explore the interface between natural and artificial quantum systems, with the goal of accelerating progress in quantum technology by learning from nature's examples. The specific mechanisms by which photosynthesis research informs quantum algorithm design operate at multiple levels, from the architectural to the procedural, and one important area involves the structure of quantum networks, for the chromophore arrangements within photosynthetic complexes represent highly optimized networks for quantum transport, with geometries that balance coherent tunneling against environmental decoherence in ways that maximize transfer efficiency. And understanding the principles that govern these natural architectures may guide the design of quantum processor topologies, informing decisions about how qubits should be arranged and connected to achieve optimal performance for particular classes of computation. Another area of influence involves the treatment of errors and noise, for as the ENACT framework demonstrates, the relationship between quantum systems and their environments is more nuanced than simple opposition, and learning to work with rather than exclusively against environmental effects represents a potentially transformative shift in approach, one that may enable quantum computations in conditions previously thought too noisy for meaningful quantum advantage, extending the practical applicability of quantum computers beyond the extremely controlled conditions that current error correction schemes require. There is also the matter of temporal dynamics. For photosynthetic energy transfer occurs on timescales of femtoseconds to picoseconds, extraordinarily brief intervals during which quantum coherence must be maintained and exploited, and understanding how biological systems manage these dynamics, protecting coherence just long enough to achieve functional benefit while gracefully transitioning to classical behavior once the quantum work is done, offers lessons for quantum computer operation, where similar challenges of timing and coherence management arise, and where biological solutions may suggest engineering approaches not previously considered. The field of quantum biology more broadly, of which photosynthesis research forms a central pillar, continues to reveal unexpected connections between living systems and quantum mechanics, from the magnetic navigation of migratory birds to the efficiency of enzyme catalysis to the sense of smell, and each discovery adds to a growing body of evidence that nature has been exploiting quantum effects far more extensively than scientists once believed, creating a rich source of inspiration for quantum technology that researchers are only beginning to fully appreciate and systematically explore. As quantum computers grow more powerful and more practical, the insights derived from photosynthesis research are likely to become ever more relevant, informing not only the fundamental design of quantum hardware but also the algorithms and protocols that make such hardware useful, and the quiet work of plants, carrying on their ancient practice of converting sunlight into chemical energy, will continue to offer lessons to those who take the time to observe and understand, reminding us that the boundary between biology and physics, between natural and artificial, between ancient and cutting edge, is far more permeable than we might initially suppose. And so as you settle further into rest, perhaps you might imagine the sunlight falling upon a leaf outside your window, the photons being absorbed, the excitons beginning their quantum walks through molecular landscapes, and somewhere in a laboratory or technology company, a researcher studying these same processes, looking for patterns that might make a quantum computer work a little better, solve problems a little faster, bring us a little closer to understanding the quantum nature of the world we inhabit, all proceeding with the same gentle inevitability as the seasons, as the growth of plants toward light, as the slow accumulation of knowledge that characterizes our species at its most patient and curious. And there is nothing urgent about any of this, nothing that cannot wait until tomorrow or the day after, for nature has been working on these problems for billions of years and seems in no particular hurry, and neither should we be, here in the quiet space between waking and sleep. And now we turn our attention to something quite practical, which is the question of how all this delicate quantum machinery inside leaves might be improved upon, or at least encouraged to work a little more efficiently, so that the plants we depend upon for food might yield more grain or fruit from the same patch of soil, and so that the forests and fields of the world might pull just a bit more carbon dioxide from the atmosphere, which is, as you may know, rather full of it at present. This is the domain of agricultural scientists and synthetic biologists, people who spend their days thinking about the inner workings of chloroplasts and wondering whether millions of years of evolution have truly optimized photosynthesis, or whether there might be room for gentle improvement here and there. At the University of Illinois, in the rolling farmland of the American Midwest, there is a research project with a name that sounds rather like a promise, which is RIPE, standing for Realizing Increased Photosynthetic Efficiency, and this project has been working since 2012 to understand whether we might coax more productivity from the fundamental process by which plants turn sunlight into food. The project is led by Donald Ort, a plant biologist who has spent decades studying the molecular details of photosynthesis, and it has received substantial funding from the Bill and Melinda Gates Foundation, amounting to some $116 million over the years, which is a considerable sum devoted to understanding the biochemistry inside leaves. The reason for such investment is straightforward enough when you consider that photosynthesis is the ultimate source of essentially all the calories that humans consume, and that any improvement to this process, even a modest one, could translate into significantly more food from the same amount of land, water, and fertilizer. One of the most striking achievements to emerge from the RIPE project was published in the journal Science in January of 2019, and it concerned a rather troublesome aspect of photosynthesis called photorespiration, which is a process that wastes a good deal of the energy that plants capture from sunlight. You see, the enzyme at the heart of carbon fixation, the one we have discussed before called RUBISCO, has an unfortunate tendency to sometimes grab oxygen molecules instead of carbon dioxide molecules, which creates a toxic byproduct that the plant must then expend considerable energy to clean up. This oxygen-grabbing mistake happens more frequently when temperatures are warm, which is rather inconvenient given that many important crop-growing regions are becoming warmer. The RIPE researchers engineered tobacco plants with a kind of biochemical shortcut, a bypass pathway that allows the plant to deal with the toxic byproducts of photorespiration much more efficiently, recycling them back into useful molecules without wasting so much energy. When these modified tobacco plants were grown in real field conditions, not just in controlled laboratory settings but outdoors in actual soil with variable weather and all the complications of real agriculture, they produced roughly 40% more biomass than unmodified plants, which is a genuinely remarkable improvement for a single genetic modification. To understand why photorespiration is such a problem, and why some plants have evolved clever ways around it while others have not, we need to consider the difference between what are called C3 and C4 photosynthesis, which are two slightly different strategies that plants use for capturing carbon dioxide. The names refer to the number of carbon atoms in the first stable molecule produced after carbon dioxide is fixed by the plant, which is a 3-carbon compound in C3 plants and a 4-carbon compound in C4 plants. Most plants on Earth, including wheat, rice, soybeans, and most trees, use the C3 pathway, which is the more ancient and straightforward approach, where carbon dioxide is captured directly by RUBISCO in the mesophyll cells of the leaf. The trouble with this approach is that RUBISCO is exposed to whatever ratio of carbon dioxide to oxygen happens to be present in the leaf, and oxygen is unfortunately quite abundant, being about 21% of the atmosphere, while carbon dioxide is comparatively scarce at around 0.04%, so the enzyme frequently makes its oxygen-grabbing mistake. C4 plants, which include maize, sugarcane, and many tropical grasses, have evolved a rather elegant solution to this problem, which involves a kind of biochemical concentration mechanism that pumps carbon dioxide to high levels around RUBISCO, so that the enzyme is bathed in carbon dioxide and rarely encounters oxygen. This pumping is accomplished by first capturing carbon dioxide in outer cells using a different enzyme that does not make oxygen mistakes, and then shuttling the 4 carbon molecules that result into inner bundle sheath cells where RUBISCO works in splendid isolation, surrounded by carbon dioxide at concentrations perhaps 10 times higher than in the air outside the leaf. This concentration mechanism has a cost in terms of the energy required to run the pumps, but in warm conditions where photorespiration would otherwise waste a great deal of energy, the C4 approach comes out ahead. The theoretical maximum efficiency of photosynthesis, which is to say the very best conversion of sunlight energy into stored chemical energy that the process could achieve if everything worked perfectly, is different for C3 and C4 plants because of these different strategies. For C3 plants, Calculations suggest a maximum efficiency of about 4.6%, meaning that at most about 5% of the energy in sunlight could be captured and stored as biomass under ideal conditions. For C4 plants, the theoretical maximum is somewhat higher, around 6%, because they waste less energy on photorespiration. In practice, actual crops achieve efficiencies well below these theoretical limits, typically around 1 to 2%, which means there is considerable room for improvement even without changing the fundamental photosynthetic machinery. This difference between C3 and C4 efficiency has motivated one of the most ambitious projects in modern agricultural science, which is the effort to convert rice, one of the world's most important food crops, from a C3 plant into a C4 plant. Rice feeds roughly half of humanity, providing daily calories for billions of people, mostly in Asia and Africa where populations continue to grow and where climate change threatens existing agricultural productivity. The C4 Rice Project, based at the International Rice Research Institute in the Philippines, has been working since about 2008 to understand the genetic and developmental changes that would be required to install C4 photosynthesis into rice plants, which naturally use the C3 pathway. This is not a simple matter of inserting a few genes, because C4 photosynthesis requires coordinated changes in leaf anatomy, with the development of distinctive bundle sheath cells surrounded by specialized mesophyll cells, as well as changes in the expression of many enzymes and the transport proteins that move molecules between cell types. The project is making gradual progress, identifying the key genes that control these developmental patterns and testing various combinations in experimental rice plants, though a fully functional C4 rice variety remains some years away. Stephen Long, a plant scientist who works closely with the RIPE project, has performed calculations suggesting that improving photosynthesis could increase crop yields more substantially than any other single intervention available to agricultural science. His analyses consider the various factors that limit crop productivity, including the availability of water, nutrients, and light, the efficiency of carbon fixation, and the allocation of captured carbon to harvestable parts of the plant like grain or fruit. While improvements to each of these factors can boost yield somewhat, the fundamental capture of light energy through photosynthesis represents the ultimate ceiling on productivity, and raising this ceiling through better photosynthetic efficiency would lift all other constraints as well. Long has estimated that combining several photosynthetic improvements, addressing photorespiration, light harvesting, and carbon fixation together, could potentially increase yields of major crops by 50% or more, which would be transformative for global food security. Meanwhile, in Germany at the Max Planck Institute for Terrestrial Microbiology, a researcher named Tobias Ebb has been taking a rather different approach, which is to ask whether we might design entirely synthetic pathways for carbon fixation that work better than anything found in nature. Ebb and his colleagues have studied the various natural carbon fixation pathways used by different organisms, because it turns out that the Calvin cycle used by plants is only one of at least 6 different biochemical routes that living things have evolved for capturing carbon dioxide. Some of these alternative pathways are more efficient than the Calvin cycle in certain respects, using less energy to fix each molecule of carbon dioxide, and Ebb wondered whether components of these different pathways might be combined into novel synthetic cycles that outperform any natural system. In 2016, his laboratory published work describing a synthetic carbon fixation cycle assembled from enzymes borrowed from 9 different organisms, including plants, bacteria, and archaea, which achieved carbon dioxide capture about 20% more efficiently than the natural Calvin cycle. This synthetic pathway, called the CETCH cycle, does not yet work inside living cells, but was demonstrated in test tubes containing the purified enzymes, and it represents a proof of concept that human-designed biochemistry might eventually improve upon evolutionary solutions. The possibility of optimizing RUBISCO itself, that ancient and somewhat clumsy enzyme at the heart of carbon fixation, has become more realistic with the development of precise gene editing tools like CRISPR, which allows researchers to make specific changes to DNA sequences with unprecedented accuracy. RUBISCO is encoded by genes in both the nucleus and the chloroplast, which complicates genetic modification, and the enzyme is assembled with the help of molecular chaperones that are finely tuned to work with the natural form of the protein, which means that modified versions often fail to fold properly. Despite these challenges, researchers are using CRISPR to introduce mutations that might improve RUBISCO's ability to distinguish carbon dioxide from oxygen, or to increase the speed at which it processes carbon dioxide molecules. Some efforts focus on replacing the native RUBISCO in crop plants with versions from other organisms, such as cyanobacteria or red algae, which have naturally more efficient enzymes, though persuading these foreign enzymes to work properly inside plant chloroplasts remains technically demanding. All of this work on improving photosynthesis has implications that extend well beyond agriculture and food production, because plants are also the primary mechanism by which carbon dioxide is removed from the atmosphere and stored in biological form. The world's plants, including forests, grasslands, and crops, currently absorb roughly 1/4 of the carbon dioxide that human activities release each year, which amounts to billions of tons of carbon moving from atmosphere to biosphere. If photosynthesis could be made more efficient, so that plants capture and store more carbon per unit of land area, this biological carbon sink could potentially be expanded significantly. Various analyses have suggested that enhanced photosynthesis in both natural ecosystems and agricultural lands could increase carbon capture by somewhere between 5 and 10 gigatons of carbon dioxide annually, which would be a substantial contribution to addressing climate change, equivalent to perhaps 10 to 20% of current annual emissions from human activities. Such estimates involve considerable uncertainty, of course, because the carbon cycle is complex and increased plant growth in one region might be offset by other changes elsewhere, and because carbon stored in plant biomass is only temporarily sequestered unless it ends up in long-lasting forms like soil carbon or wood products. Nevertheless, the possibility of engineered plants that grow faster, produce more food, and remove more carbon dioxide from the air is genuinely appealing, and it has attracted attention from governments and foundations concerned about both food security and climate change. The research continues in laboratories and experimental fields around the world, slowly accumulating knowledge about the genetic levers that control photosynthetic efficiency, and gradually developing the tools to adjust those levers in useful directions. And so we see that the quantum mechanical processes inside chloroplasts, those delicate movements of energy through precisely arranged molecules, are not merely subjects of academic curiosity but have practical implications for the future of agriculture and the composition of the atmosphere itself, and that scientists are working patiently to understand and improve these ancient biological systems, applying the tools of modern molecular biology to enhance processes that evolved billions of years ago, in the hope of creating crops that yield more food and ecosystems that capture more carbon, all powered by the same reliable sunlight that has sustained life on Earth since the first photosynthetic organisms appeared in the ancient seas.